Let's get started. Good morning. I'm Wolfram Goessling. I'm a liver doctor and the chief of gastroenterology at Mass General. And together with my colleague, Sangeeta, Bhatia, I wanted to welcome you all to our 33rd annual Arias Symposium. The symposium was conceived at a significant birthday for Dr. Arias 33 years ago. And we are all very excited that Dr. Arias is here to join us for this symposium as well. It's really designed to bridge basic science and liver disease. But for us, Sangeeta and I, we've been have been organizing this meeting since 2017. We brought it here in the center of innovation and biotech in Cambridge at the Broad to really build local community of those who are dedicated and interested and fascinated by the liver and liver disease. And over the years, I think we have been able to grow this community. We've weathered the pandemic and moved to hybrid for a few years and to full remote for a while. And now we're back for the second year to have this in a hybrid format and to welcome you all here in the auditorium and all of you wherever you are in the world online. This year, we have more than 450 registrants overall. We have or expect 150 here locally. We have 10 fantastic and distinguished speakers from all around the world. Dr. Francis Collins will kick us off in a moment. And we have trainees, representatives from our local universities and biotech, as well as here as speakers. I wanted to personally, before I hand it over to my colleague, also highlight that for the 7th year that we have been organizing this, we made a commitment in the beginning to have equal or strict gender balance with our speakers. And as you look at today's program, as every single one in the previous years, we have equal participation of male and female scientists. And I think that is something that not every symposium achieves, even these days. And so with that, I'll hand it over to Sangeeta.
Thank you, Wolfram. Hi, everyone. I'm Sangeeta. It's great to see all of you. And as Wolfram said, this is our 7th year doing this, and we've been really excited to grow the community and in particular to show trainees who are thinking about careers in science, medicine, and biotech that the liver is absolutely what you should be paying attention to. We think it's the center of the universe, and hopefully you will too by the end of the day. We have 7 trainee micro talks today and 19 posters that you'll see over lunch. So we have the wonderful plenaries happening, but also there's lots of emerging science that you'll get exposed to. Before we go any further, we wanted to thank Heather Fleming, who actually, is she here? She's, Heather, we're thanking you. Yeah. She really pulls this off almost single-handedly together with ALF every year, and we're so grateful for her tireless efforts. You'll see her fingerprints on everything that happens over the course of the day. So with that, we have a really fun day ahead. We're going to talk about a variety of topics. From Dr. Collins, we'll hear about efforts to finally eliminate HCV, the role of the immune system in transplant and immunotherapy, understanding the cell identity of healthy liver regeneration, as well as dysregulated growth in pediatric liver cancer. And then we have a bunch of enabling technologies that may or may not be in the liver, but we think could be applied to the liver. So you'll hear about multi-scale and spatial imaging, the role of single-cell data in understanding drug toxicity, and also the development of RNAi therapeutics. So we're really excited for the diversity of topics that you'll all be hearing about. And some of you are coming from AASLD and know that it's actually been a really exciting year for the liver clinically. We have the first approved MASH drug there. I said it, MASH. And this year we had 2 hepatocyte-targeted CRISPR therapies in the clinic. We have advances in diagnostics and liquid biopsy for HCC. And we have the recognition in the wake of COVID that the lipid nanoparticles that were delivered to so many millions of patients for mRNA delivery, when administered systemically, naturally go to the hepatocyte. And that has gotten many, many people excited about targeting the liver, and then also conversely detargeting the liver if you want to go somewhere else. So we have many people joining our community who sort of traditionally weren't here, which is wonderful. So with that, sit back, enjoy the day. If you make a friend, a collaboration, learn something new, we consider that success. And with that, I'm going to hand it off to Vardman to get started.
Hi, I'm Christina Lopez. I'm a graduate student in Kristen Knaus's lab.
Hi, I'm Vardman. I'm a postdoc researcher in Sangeeta's lab.
We'll be your co-chairs for the first session. Some quick reminders. I think on the next slide, there will be a QR code that has the agenda for the day. For those on Zoom, please ask your questions in the Q&A panel. Anything else?
And there's a break after the first session, so the speakers for the second session can use that break time to test their AV and presentations if you want. With that, it's our honor to have Dr. Francis Collins presenting with us here. He doesn't really need any introduction, but just to keep it brief, Dr. Collins is a physician-scientist, and under his direction, the Human Genome Project produced the first finished sequence of the Human DNA Instruction Book in 2003. From 2009 to 2021, he served under 3 presidents as the director of the US National Institute of Health. Following a year in the White House as president's acting science advisor, he oversees a research laboratory as a distinguished investigator in the intramural program of National Human Genome Research Institute. He also leads a bold administration initiative to eliminate hepatitis C in the United States, and we are excited to hear about that in his talk. His contributions to science, medicine, and society have been recognized by the Presidential Medal of Freedom, the National Medal of Science, and the Templeton Prize. And his most recent book is The Road to Wisdom on Truth, Science, Faith, and Trust. And with that, Dr. Collins, we are excited to hear from you.
Well, thank you very much, co-chairs, for that kind introduction. And thanks to Wolfram and Sangeeta for organizing this over these past several years. And I'm honored to be part of it and wish I could be there with you in person, but other things are preventing that today. But glad that I can join you virtually. I will have some slides that I can show, so maybe I will try to put those up and hope that they appear in the appropriate way. So let's see how that looks. So I'm unable to see you all, but I'm assuming that this is coming through, or somebody would tell me otherwise.
Yes, we can see the slides, Dr. Collins.
Thank you, that's great. So I do want to tell you about a bold national initiative to eliminate this now curable disease in the United States. But I also want to say what a privilege it is to be part of something that recognizes my friend, Win Arias. Win and I've known each other quite a long time. He runs the program at NIH called Demystifying Medicine, which brings together basic scientists and clinicians, just as you are doing now here in this series of meetings, now the 33rd annual, to do the bridging between the basic science of the liver and its clinical applications. I serve as the White House lead on this initiative. I got engaged in this when I was asked to be the president's acting science advisor back in early parts of 2022. I had stepped down as NIH director, thought I was going to have a quiet year. Didn't quite turn out that way. One of the things you can do as the science advisor to the president is to look across the entire government and see, are there things that we could be doing more about that haven't quite come together? And having already had an interest in hepatitis C before that, I focused on this fairly early on, and it seemed really compelling that while we have seen one of the most amazing achievements of the 21st century with the development of direct-acting antivirals for this very serious disease, they were not reaching a lot of the people who needed them, and that seemed like something that we ought to work together to try to achieve. So I'm going to tell you about that. I know we have a diverse audience. Many people are intimately familiar with hepatitis C, others not so much. So I'm going to provide a little background, but I want to focus primarily on where we are at a critical juncture here of seeking to launch a major initiative that would aim to try to find, test, treat, and cure as many of those who are currently infected with this virus in the United States, and to do so efficiently in a way that actually saves lives and saves money. The epidemiology of hepatitis C is caused by the transmission of a virus through contaminated blood. The peaks that you see here in CDC's description of where the cases are reflect that. The baby boomers, most of those are individuals who acquired the virus through blood transfusion before we knew how to screen to find those units that were potentially risky. And because of the very long incubation time, while we've screened since 1993, there are people who got blood before that who may just now begin to start to show symptoms, and there's a lot of them. There's less in Gen X, but the millennials, which is the fastest growing group, primarily these are individuals who got infected by a shared needle in the process of intravenous drug use or perhaps a dirty tattoo needle. And that, because of the opioid crisis, is an area of considerable concern. If you do hepatitis C testing for individuals who are currently undergoing treatment for opioid addiction, you will find a very high proportion of those in some programs, as much as 50% that are hep C positive. And that is a group that at the present time, we have not reached nearly as effectively as we should. 40% of people with hepatitis C don't even know they have the infection, while CDC and USPSTF have made recommendations about how to increase our screening attention. CMS has not recently upgraded their recommendation, and there's still a failure to do the kind of screening that ideally ought to be done to discover those who are infected, especially because there is now a cure. The natural history of the disease stretches out over 2 or 3 decades in many instances, without much in the way of symptoms for the first many years of this. But over a course of time, the virus in the liver leads to hepatitis, gradual fibrosis, and ultimately cirrhosis, which can then present with symptoms that can be quite significant, including bleeding episodes, including fatigue, and unfortunately also a high risk of liver cancer. This is now the most common cause of liver cancer in the United States, and it's a cancer that doesn't respond particularly well to therapeutics. So this all sounds pretty grim. The good news is this is a curable disease. Remarkable research done by initially academic investigators, but then taken up with great success in the pharmaceutical industry, has brought us to the place where we have now direct-acting antivirals that are highly effective, safe, and lacking much in the way of side effects, the kind of thing that you wish for in many diseases, and now we have it for hepatitis C. The life cycle of the virus when it enters the hepatocyte involves uncoding, and then it makes a single RNA of about 3,000 amino acids that then has to be cleaved by protease. And then there's a variety of other steps involved, all of which involve these non-structural, that's what the NS means, genes in the viral genome. And so it's a great opportunity then to develop targeted small molecules that block these various steps, because if you get a very specific small molecule, you should be able to have both high efficacy and very low in the way of side effects because you're not hitting a host target. It is a very good thing that this has been investigated thoroughly. And so there are, in fact, inhibitors against 3 different areas of the life cycle of the virus. And as you might guess, if you're trying to cure a viral disease where the viral load may be pretty high, it is to your advantage to be able to come at this with more than one drug in order to avoid the likelihood of the arrival of resistance. Same arguments that mean we treat HIV/AIDS now with 3 antiretrovirals. Here for hepatitis C, the current drugs combine at least 2 of these direct-acting antivirals and are highly successful. Just want to emphasize for a minute, because this is a meeting that's talking about the importance of bridging from basic science to clinical applications, that this is really a masterpiece of that kind of translational research. I have a quote here from a recent, really elegant review of hepatitis C in Nature Reviews. Let me just read it to you. The history of HCV research reads like a role model for successful biomedical and translational research, starting from a clinical observation, non-A, non-B hepatitis, via identification of the underlying etiology, a virus, the establishment of diagnostic tests, unraveling of the viral life cycle, development of specific therapeutic agents, and finally, implementation, yet to be fully achieved, of a global elimination program. The HCV story is a masterpiece of translational research. I totally agree with that, and was honored, of course, by the Nobel Prize in Physiology or Medicine in 2020 for Harvey Alter, Michael Houghton, and Charles Rice. So yeah, as we consider ways in which we could really build these bridges between basic and clinical, this is a great model to try to follow. Now, what about that global elimination program? Well, it is certainly a goal that has been set by WHO by 2030, and some programs in some countries are actually moving along pretty well. This is just a sample of those. The UK has been working on this. Egypt kind of jumped out ahead of everybody, and if you read that report in the New England Journal, essentially achieved success in finding and curing individuals with hepatitis C for the whole country. The country of Georgia has a program that's been going on for a while. Australia has made wonderful progress, and we've actually been learning quite a bit from their progress in a particular way in terms of how they have set up their testing and treatment. But if you want to read a more thorough analysis of where we are as far as global elimination, I'll point you to this article, which just came out a week or so ago, and is available online in the Annual Review of Medicine, a summary of the status of all this. The first author, my senior advisor, Dr. Rachel Florence, but you can also see other authors here, Harvey Alter himself, the Nobel Prize winner, and John Ward, who's been leading the global effort very effectively from his position at Emory University. But what about the US? Why, in fact, have we not succeeded at this? The first approved antiviral, direct-acting antiviral for hepatitis C was 2011. That one still required the use of interferon and ribavirin, which are pretty toxic. So it was more like 2014 that we began to see purely oral agents with high success rates appearing. But we still have these 4 million currently infected individuals in the United States who have not reached that point of a cure. Reasons for that. This CDC diagram of the hurdles that are being faced covers quite a lot of that, and there's a bunch of them. Lack of awareness. You're not going to cure people if you don't know they have the disease. The diagnostic algorithm, which until recently has required something like 3 visits to the clinic before you start therapy. And we lose a lot of people along the way because people who are infected with hepatitis C in the early years don't have symptoms. Many of them may be limited in terms of access to care or transportation. And the idea of you got to come back 3 times before you even get started on treatment is not the way you'd want this to be. The initial approach then was an antibody test to see if you have evidence of infection, but recognizing that about 20 to 25% of people with hepatitis C are able to cure themselves. That is a possibility. Then you want to know, do they have active infection? That requires a viral RNA test in peripheral blood. Both of those tests, the antibody and the RNA test, generally have to go off to a central laboratory. It takes time to get the result back. And so only in that 3rd visit, if both of those tests were positive, is it time to start therapy. Other things that get in the way, the high cost. The original cost of a 12-week, 1 pill a day treatment was $94,000. That has come down with competition now. Both Gilead and AbbVie have direct-acting antivirals that work against all genotypes of Hep C. but the cost is still around $24,000. That meant that in circumstances where resources were tight, like state Medicaid programs, or certainly in community health centers that are treating the uninsured, or in the prison system where hepatitis C can be as frequent as 20 or 30% of the residents of many prisons and jails, there just wasn't the resources around to be able to do this. And so there was a reluctance even to do the testing because you weren't sure that you would be able to actually offer the cure that people need. Because of that, various barriers got put up, requiring prior authorization, requiring a specialist to see the patient. I'm an internal medicine doctor. I can tell you that the testing and treatment and cure of this disease is pretty simple and pretty gratifying because it works 95% of the time. So the need for a specialist in most instances, frankly, is not very compelling at all. Primary care docs, nurse practitioners ought to be able to do this quite effectively. But then there's this big issue about underserved and hard to reach populations. A lot of the younger group that is infected with hepatitis C are people who were involved in some way in intravenous drug use or may currently be so. And as a result of that, many of those individuals end up in difficulties with the justice system and may therefore also be difficult to reach. But if we don't succeed in doing that, these individuals will end up with serious consequences from liver cirrhosis and liver failure and needing liver transplants or treatment for liver cancer. It is simply not acceptable to say, well, that's okay, when you consider what the consequences are, both on a compassionate basis and, as I'll mention in a minute, on an economic basis. So how are we actually doing in the US about this? Pretty badly, actually. This is CDC's data from about a year ago. These are people who you know have the diagnosis. They did get tested. They're in the 60% where you know they have the virus. And yet, over the course of a year after their diagnosis, only 1 out of 3 is getting cured. And it's worse than that if you have no health insurance or you're young or both of those. This is simply not an acceptable circumstance in a situation where we could do so much better. Now, some would say it's just too hard reaching out to marginalized communities of this sort, so we just have to accept this. No, that's not the right answer either. As a counterexample, I will tell you the Veterans Administration, having received a significant input of funding from the Congress, mounted a very successful hepatitis C elimination program, and were able to reach out and find something like 85 to 90% of people and get them tested and get them cured. It required a significant amount of funding because of the cost of the drug, but they did it. And this tells you that this is possible. And furthermore, various states have mounted pilot programs, most notably Louisiana, where in that instance, they targeted individuals on Medicaid and people in the federal prison system. They came up with a strategy to make the drug more affordable. Basically, they worked out what's called a subscription model, or sometimes called a Netflix model, with Gilead, one of the drug suppliers. Gilead was willing to accept a lump sum upfront of what their payment would be for access to the drugs. And then once that had been decided, the drugs could be made available to anybody on Medicaid in Louisiana or anybody in the prison system without further cost. So there's a big impetus then to go and find everybody who's Hep C positive. The next person that you treat doesn't cost you another dime as far as the therapeutics because you already paid for that. It's a very attractive model and one which we now want to take to the national level. Everybody wins that way. The company is well compensated. It turns out it's very cheap to actually make the drugs, so the cost of supply is quite limited. And certainly in terms of finding and curing people, that is the goal that you would have. This supports that. Louisiana increased in the very first few months after initiating this program, their cures of this disease 5-fold. So if that's all possible, why are we not acting on this? Some of it is just inertia. As the as the White House Science Advisor, talking to lots of different parts of HHS and the prison system, it just seemed like we just need the momentum to get this going. But that's not a trivial circumstance. Will require budgeting and congressional authority and able to make that happen. I've been pretty noisy about this. I'm a little bit obsessed about it, to be honest. How can you have this kind of major medical achievement and not have it get to the people who need it? I wrote an editorial about this about a year ago in the New York Times, using strong language. We're squandering one of the most important medical advances of the 21st century. How can we do this? Well, let me tell you where we are, because I think we are on the cusp of what could be a major success story, but it's not quite in the bag at the moment. Working over these 2.5 years with lots of input from people on Capitol Hill and in the executive branch, and particular leadership, I might say, from 2 senators, Senator Bill Cassidy of Louisiana, who happens to be a liver doctor, and Senator Chris Van Hollen of Maryland. That's a Republican and a Democrat. This is a bipartisan effort, and with a lot of support from the White House and personally from President Joe Biden. Here's what the plan is that has been put together, some of which I'm glad to say has already been accomplished. First of all, we needed to have a better testing parameter system so that you didn't have this long delay in figuring out whether somebody's infected and getting them started on treatment. That means a point of care test that can be done in less than an hour, allowing test and treat in a single visit. The point of care test would have to be for viral RNA, so you know that somebody is currently infected. That kind of test has been available in other countries, Australia and some European countries, but was not available in the US. FDA requires a very stringent system to be able to test out this kind of advance. And so working with FDA, NIH, through a program called RADx and a program called ITAP, was able in a very short period of time to set up a clinical testing program and show that this particular finger stick test, which is provided by Cepheid, had very high sensitivity and specificity. And this was authorized and approved by FDA this past June, really in record time. So that means that in low resource settings, for instance, like opioid treatment centers, where you may not have a phlebotomist or a physician necessarily on site, you have the opportunity to do test and treat of individuals in a single visit. This is a game changer for a lot of the things that we need to implement. So that was point number 1. Point number 2 was to be able to do something about broad access to the curative medications. Again, building off of the success of the Louisiana pilot, and some pilots in other states have also tried this. We think this is the right way to go for the whole country. Negotiate with the drug suppliers, and the 2 companies that have drugs that cover all genotypes are AbbVie and Gilead, for a reasonable price to cover their need for making a reasonable profit on these groups. But basically then make the drugs available at that point for free to individuals in Medicaid, individuals that are uninsured, people in the prison and jail system, and Native Americans cared for by the Indian Health Service, where hepatitis C is also increased in its frequency. The deal here then means that the actual cost per patient goes down a lot because you have a lot of patients, but the company's profits also are maintained. On top of that, simply having availability of the drug is insufficient. We have to boost up the healthcare delivery system to make it possible to reach high-risk populations. That's going to involve a lot of telehealth. There are great details underneath this slide that I don't have time to tell you about that have been implemented and put together into a very carefully planned package that includes now a 35-page piece of specific legislation that has been assembled by Cassidy and Van Hollen and other members of the Congress. So that's the plan, and that is very much now out there waiting for the opportunity for initiation. And of course, the question is, where is it at the moment? Let me walk you through in this pretty much last slide about where we are. Initially proposed as part of the president's budget in March of 23, so the proposal's been out there. There is strong federal agency support across all of HHS, particularly from CDC, from HRSA, from SAMHSA, from the Indian Health Service, and from the Bureau of Prisons. Everybody agreeing we can do this. A detailed plan prepared about how to do this in 5 years and reach out to as many of those 4 million people as we possibly can in that timetable. The advocates are organized, and a big shout out here to AASLD because they've led an advocacy organizational effort to get letters to the Hill and pound the halls of Congress to point out what a remarkable opportunity this is that should not be missed. The legislation has been developed, not yet formally introduced, but I would say pretty much finalized. There is bipartisan interest in the Congress. I've personally talked to at least 30 members of Congress to explain what this could be and found pretty much universal interest, but obviously a big question about the cost, which is what I'm coming to right now. What about the cost? Well, think about this. If you find these people and get them cured, then they don't end up with liver cirrhosis and needing a transplant or treatment for cancer. And you can calculate, based upon the natural history of this disease, what that adds up to. Our own economists, particularly Jag Chatwal at Harvard, had done that analysis over the last couple of years, and it looked very encouraging in terms of cost savings. But nobody believes the analysis until it comes from the Congressional Budget Office, the CBO. The reductions in fibrosis, cirrhosis, cancer, transplants, and also diabetes and kidney disease, which turn out to be a consequence of chronic liver inflammation from this virus and can be reduced by cure, ends up adding up to quite a lot. Just back in June, CBO predicted this would save, if we could just double the number of Medicaid recipients getting cured, $7 billion in 10 years. We actually think we could probably triple that number based upon the analysis that's been done by CDC and other agencies. CBO continued to do their full cost analysis, which counts all kinds of consequences here, including such things as people get cured, and so they might need Medicare later when they get older. You have to put that in there too. They signaled a month ago that this effort would show savings in 10 years. It was going to be a cost saver. That's a big deal in terms of getting something through the Congress. And their final score has just been sent to the Congress. I'm not in a position to tell you what the number is, but it's really good. So here's a circumstance where you have a bill that could save lives and save money. And in congressional language, that means if there are other things that need money to make them happen, like a telehealth, for instance, that this is an opportunity for the pay-fors that come out of hepatitis C to help other things. So the big question now is, can this all come together in some sort of end-of-year package, which needs to happen in about the next month, in order to get this done, signed into law by the president and initiated in the early days of 2025. That is, in fact, my hope. And there's a whole lot of other tireless people working with me, my small but mighty team of Rachel Florence, my senior advisor, Josh Sharfstein from Hopkins, who's thrown himself into this in a big way, Risha Irwin, also from Hopkins, and Kevin Klinkenberg, who works for HHS legislation, have been 100% devoted to getting this through. And again, we've had lots of support from the executive branch, but also from the Congress. So bottom line, unless we take action, we're going to keep spending tens of billions of dollars for care of people over the coming decades as their livers fail. Why don't we invest now in a cure? It's compassionate, prevents suffering, and save tens of thousands of lives. I watched my brother-in-law die of this disease 10 years ago, and it was a really awful outcome. And it's also economically wise. This is long-term deficit reduction. So I'll stop there and see if there are questions that people want to pose, although I know we got started a little late, so I will count on the organizers to decide what to do about the question part. And just to say, I'm not hearing anything. So if there's some switch that needs to be flipped so I can hear something.
Oh, can you hear?
Now I can hear you, yes.
Oh, so that was terrific, Dr. Collins. Thank you. In the clinic, in issues now arising that we didn't, so when we were treating these patients 10 years ago, starting to treat them, if they had less than F2 fibrosis, we cured them, we sent them off and said, have a great life. You're all set. Higher degrees of fibrosis, we figured we should screen for hepatocellular carcinoma. Now, 10 years later, we're starting to see these patients who we thought were all set showing back up with rather aggressive hepatocellular carcinomas, which were unanticipated, and we didn't screen. And so I guess the question here is there's no doubt that we would reduce the overall risk of HCC by this program. The question is, the rate of this seems to be non-trivial. It may be an epigenetic effect of the virus. It's unclear, so that, it's a point of study. But I guess the question in this program, should there be some connection to care that, that's fit in at the end of, of the protocol that you presented?
Yeah, I definitely think that's an important issue. How frequently this kind of late result in somebody who got treated early is going to happen is not that clear, at least not to me. There is a group of liver experts, part of the scientific community, that is revising, though, the algorithm of what really ought to happen in the circumstance where we have a point of care test and treat can be done in one visit. That needs to still include the possibility of follow-up over the longer term. What we don't want, though, is for a lot of obsessing to happen about trying to get everything perfectly arranged before you start the treatment. The goal here really is to find people as quickly as possible and get them started on treatment and work out whatever needs to happen once they've achieved SVR in the longer term, because we've waited far too long to make this big push to just go and make those cures happen.
Francis, this is Wynn. Thank you very much for being with us. We appreciate it very much. What is the status of competition within the pharmaceutical industry for derivatives or other modifications of the drug? Do the patent laws permit that sort of thing? And is there the possibility of competition that would really bring the prices down?
Well, right now, I think because there are two very well-established FDA-approved direct-acting antivirals that work on all the genotypes, which is something you want because you don't want to have to do genotyping of each patient. That's Gilead and that's AbbVie. I'm unaware of other pharmaceutical companies that are seeing this, therefore, as an attractive place to make an additional investment. But there may be that going on, and I just don't know. The current patents, though, on the existing direct-acting antivirals are going to be in place for at least the next 6 or 7 years, maybe longer. So in terms of what some have said, well, let's just wait until the patents expire and then this won't be so expensive. That's waiting way too long. 40 people will die today of hepatitis C. 15,000 will die each year for a curable disease. So every day that goes by that we haven't started this, feel like we're missing an opportunity to save lives. So as much as I would love to see some other competition drop the price further, at the moment, it doesn't seem like it's likely to happen real soon.
Tina, are there any questions on Zoom?
Hello? Does that work?
Yeah.
All right. Question from Zoom is, what about insurances that still do not cover this treatment? What efforts are being done on that end?
Well, that's an important issue. It's certainly the case that people with third-party insurance may still encounter barriers that are put up to getting access to the treatment because the insurance company doesn't want to have to pay for it. And by the way, the proposal in the bill is to cover with this subscription model 4 groups: Medicaid, uninsured, prison system, and Native Americans cared for by IHS. It does not affect third-party insurance. It does not affect Medicare, except we do propose dropping any co-payments in Medicare because they have been sometimes a deterrent to people getting cured. But for third-party insurance, I think the pressure of public influence ought to be substantial here about the failure to really justify why you would not, in this circumstance, immediately when somebody tests positive, get them treated and skip all of these other barriers involving specialists and so on. But that's not included in the bill. That's going to have to be more of a public demand. One of the hopes I would have is if this initiative gets started as soon as next year, it will do a lot to raise public consciousness. We will have a better chance of more universal testing so that people really know if they're infected. And even people who are on Medicare right now, who maybe have delayed getting a chance to look at this, will do so as people with third-party insurance. We'll just boost this up. And by the way, that will make the companies very happy because they will end up in those circumstances where the prices have not really going to change, do pretty well. Again, the goal here is to make this a win-win-win. A win for the people who are going to get cured, most importantly. A win for the nation by saving us a lot of money, reducing our deficit by billions of dollars. But also a win for the companies who want their investment, which has been phenomenal here to achieve this kind of outcome, to be recognized.
All right. Thank you so much for joining us, Dr. Collins. Can we give another round of applause for Dr. Collins?
Thank you all. Great to be with you. Congrats to Win. 33 years and counting.
Right. Our next speaker for today is Dr. Udeme Ekong. She's a professor of pediatrics and surgery at Georgetown University School of Medicine, Washington, D.C., and an attending physician in pediatric transplant hepatology at MedStar Georgetown University Hospital in Washington, D.C. Dr. Ekong attended medical school in Nigeria and proceeded to the UK and US for residency and fellowship, training in pediatrics, pediatric gastroenterology, and pediatric transplant hepatology. In addition to her clinical work, Dr. Ekong is an active researcher. Her laboratory interest is in mechanisms that underlie regulatory T-cell dysfunction in autoimmune and alloimmune liver diseases after liver transplantation. She's looking into the molecular mechanisms behind regulatory T-cell dysfunction as a result of inflammasome activation, human endogenous retrovirus reactivation, and ER stress. The laboratory also has a strong history of collaborative work with collaborations at the NIH, Yale, Columbia, and Imperial College London. With that, I would invite Dr. Ekong to give a talk.
Good morning once again. So I'm a pediatric transplant hepatologist, meaning I look after children with liver disease and who get a liver transplant. And I'm going to be talking today on a condition called post-transplant alloimmune hepatitis that we see in individuals after a liver transplant. I have no disclosures. This is the outline of the talk I'm going to be doing today. I'm going to start off with an introduction, just giving some background on what this condition is, recognizing that not everyone here is a transplant hepatologist. I'll be talking about specifically what it is and why folks like me think it's probably a good idea to try and know a bit more about it. And then I'm going to very quickly highlight some of the past published work from our lab. We've really been focused on regulatory T cells and how the role it plays in disease pathogenesis. And I really want to spend the majority of my time talking about unpublished ongoing work in our lab at the moment. So post-transplant alloimmune hepatitis is a poorly understood cause of late allograft dysfunction. It shares biochemical and histological features with autoimmune hepatitis that's actually seen in the native liver. And if you look in the literature, you're going to see single center reports that talk about a frequency of maybe 5 to 10% in children and 5 to 10% or 3 to 5% in adults. But its cause and underlying pathogenesis remain unclear. And so how do we make a diagnosis of these? These are usually children or individuals who are transplanted for diseases that do not have an underlying immune basis. What do I mean by that? So these are folks who have things like cystic fibrosis, alpha-1 antitrypsin deficiency, the familial intrahepatic cholestasis syndromes, Alagille syndrome. So these are genetic type diseases that have no real immune underpinning. And these individuals get a liver transplant and following that develop this post-transplant alloimmune hepatitis. Most cases present at about a year after transplant. And some of the ways we go about to make a diagnosis is we look at autoantibodies. These patients have increased titers of autoantibodies. So these are anti-nuclear antibodies, anti-liver smooth, liver kidney microsomal antibodies, smooth muscle antibodies. They also have elevated levels of serum immunoglobulin G in their blood. And when we do a liver biopsy, which we always do to make a diagnosis, the portal tract of the liver is expanded with a ton of inflammatory cells. These inflammatory cells are predominantly lymphocytes and plasma cells. And when these inflammatory cells go beyond the limiting plate of the portal tract and surround the hepatocytes, they have this a finding which pathologists call interface hepatitis. And importantly, there's the absence of any other cause to explain why that individual has graft dysfunction. And so this is just a typical example of a liver biopsy with features of alloimmune hepatitis. So this is a portal tract. The portal tract is typically made up of a bile duct, portal vein, and a hepatic artery. I don't have a hepatic artery on this, but suffice to say, all of these blue are inflammatory cells, lymphocytes, and plasma cells. And when they go beyond the limiting plate, pathologists tend to call, kind of surrounding the hepatocytes, pathologists tend to call this interface hepatitis. Now, we wanted to know, if you think about it, for us in pediatric hepatology and transplant hepatology, 50% of our children are getting transplanted less than the age of 2, so they have a whole long life ahead of them. And so we wanted to know, what are the long-term consequences of a child developing post-transplant autoimmune hepatitis in their transplanted liver allograft? And to try and answer this question, we took data from 5 pediatric liver transplant centers. Some of these are centers that I worked in, which made it easy for me to work to get them to do what I wanted them to do. 4 of these centers were large centers, and 1 Yale was a smaller center. And so this was Birmingham Children's in the United Kingdom, Sick Kids in Toronto, Lurie Children's at Northwestern in Chicago, the Morgan Stanley Children's Columbia in New York, and Yale New Haven. And essentially what we did was, in each of these centers, we identified all children who had a diagnosis of alloimmune hepatitis, and then look through their charts to follow what happened to them, really from diagnosis to either death or liver transplantation again, or transition of care to an adult institution. And we use the standard definition for alloimmune hepatitis. And just to give you some background, we had a denominator of about 1,800 children, of which 31 of these children had a diagnosis of alloimmune hepatitis. The median duration after transplant when they developed this was about 5.3 years. And we had complete data for only 29 of these, follow-up data for 29 of these patients. And the median follow-up from diagnosis was about, median follow-up after diagnosis was about 7.1 years, and as far out as 15 years. And so just the pertinent findings I want to share is really, it struck us that even at the time of last follow-up, many of these patients were actually still receiving quite significant immunosuppression. And what do I mean by this? About 2/3 of them were still receiving steroids. We usually try to get you off steroids by 3 months after transplant. 3/4 were getting a supplemental immunosuppression with an anti-metabolite, and 7% were also on an mTOR inhibitor. And despite all of this, about 38% of these children continued to have evidence of liver perturbation because they had continued liver enzyme elevation. And in fact, the continued perturbation of the liver led to development of end-stage liver disease, and 14% of these children developed portal hypertension. And at the time of the manuscript submission, 10% had been relisted and retransplanted. Indication for retransplant was chronic rejection and portal hypertension. And this is just the explanted liver for these patients, and this is just a cirrhotic. This is a kid from Sick Kids in Toronto. This is just a cirrhotic nodule, deeply stained with bilirubin, as you can see, and this is scar tissue. And this is the H&E, showing us, again, in the portal tract, we see an artery, but we do not see any bile ducts within this portal tract, typically representative of chronic rejection or ductopenia. This just shows, again, this is a trichrome stain showing the staining of scar tissue. This is a cirrhotic nodule. And you see the absence of a decent size bile duct, again, suggestive of chronic rejection. This is the same thing from a patient from Yale, cirrhotic nodule, scar tissue, deeply staining yellow with bilirubin. This patient had a lot of inflammation at the time of retransplant. And we see again a cirrhotic nodule with scarring and the absence of bile ducts of chronic rejection. And so just to summarize, our observation was one of continued abnormal elevation in liver enzymes, raising the question in our mind, are there other pathways that were driving disease pathogenesis that our current medications are not targeting at this point in time? Also, as a consequence of this persistent liver enzyme elevation, these patients developed chronic rejection, end-stage liver disease, portal hypertension, needing another transplant, again underscoring in our mind the inadequacy of our present treatment options. Now, in our laboratory, we've taken the tact of actually focusing on regulatory T cells, and this is just based on the role regulatory T cells play in maintaining tolerance to self-antigens, in curbing autoimmunity, and the fact that they do play defective Tregs play a role in breakdown of self-tolerance. And in some of our very earlier work, we've looked at the regulatory T cells of these patients, the circulating regulatory T cells in blood, and we've been able to describe that these regulatory T cells seem to be pro-inflammatory, cytokine producing, and they do tend to have this Th1-like phenotype. That's actually driven by monocytes in an IL-12 dependent manner. We've also shown that these monocytes from these monocytes from these patients undergo there's inflammasome priming of these monocytes that's mediated by toll-like receptors 2 and 4, and that's really truly what's driving these monocytes to produce IL-12 and drive these Tregs towards a Th1-like phenotype. We've also recognized and reported on this, the Tregs also have a Th17-like phenotype, and we've asked the question, what is driving this differentiation to Th17-like Tregs? And we've also reported that there are these human endogenous retroviruses that are inducing unfolded protein response activation or UPR activation in these Tregs. And we think that this may be a mechanism, an additional mechanism for regulatory T cell dysfunction. Human endogenous retroviruses, as you well know, are represented about 8% of our human genome. And so essentially, we have evidence of unfolded protein response activation as a result, evidence by transcriptional upregulation of activating transcription factor 6, or ATF6, and cleavage to its active form. Now, unfolded protein response activation occurs in response to endoplasmic reticulum stress. And so we know that we've shown that there's an interaction between ERV and ATF6. Immune hepatitis, but we've also seen this in transplanted children who have autoimmune hepatitis. And we know this is true because when we do silence these human endogenous retroviruses, we get down regulation of ATF6 alpha, and these Tregs, which had a poor ability to suppress effector T cell proliferation, do recover some, not 100%. They do recover some suppressive function of Tregs. And we think that together all of this suggests that ERV1, by inducing UPR activation, may contribute to failures in immunological tolerance by lowering the threshold for pro-inflammatory signaling in these Tregs. And we think, therefore, that there's involvement of ERVs in the pathogenesis of post-transplant alloimmune hepatitis, as well as autoimmune hepatitis in non-transplanted children. So now I just really want to spend the rest of my talk just sharing or highlighting some of the ongoing work, ongoing thoughts of things that we're doing in our lab. Now, it's important. A lot of what I've said has really just been looking at Tregs, circulating Tregs in the blood, but it is important for us to also look at what's going on in the liver as well. And we have also shown, this is just RNA-seq of formalin-fixed paraffin-embedded liver tissue. We have shown upregulation of this is actually the ERV-S71. The new nomenclature for this is ERV-T in the liver of children who have alloimmune hepatitis. And this is in comparison to similarly transplanted children who have normal graft function. And this is just a confirmatory QRT-PCR showing again significant upregulation of ERV-S71 or ERV-T in the livers of children with alloimmune hepatitis compared to similarly transplanted children with normal graft function. And one of the questions that's always asked is it the inflammatory microenvironment of the liver that's actually causing activation of these ERVs? And so as another study group, we've included similarly transplanted children who have acute rejection of their liver allograft, because acute rejection, the liver with acute rejection actually has an inflammatory microenvironment, but we can see that we're not seeing upregulation of ERV. Now, the rate-limiting step to doing and is to trying to study the liver in patients is really number of cells. As you can imagine, the liver biopsy for us and our children is really literally a string of tissue. And, you know, you isolate intrahepatic lymphocytes, you'll be lucky, you get 50,000 or so cells. And so what we are doing right now is really trying to lean into the IBEX knowledge base in Ron Germain's lab at the NIH, where to try and really answer a lot of very, very important questions, some of which are on the slides, but importantly, which cells are actually upregulating ERVs in the liver. And I think it's important. I certainly, for one, would like to see, know a lot more about the spatial organization of the liver microenvironment and how that may, putting on my clinician cap, how that may relate with clinical outcomes in our patient. Now, while that's ongoing, the additional thing that we've done is we've been successful in expanding enriched CD4 cells from intrahepatic lymphocytes obtained from liver biopsies under Treg polarizing conditions to Treg. So these essentially are enriched CD4 cells that are cultured with recombinant IL-2, TGF-beta, CD3, CD28, dynabeads for about 14 days. And this is what they look like in culture. And we've then subjected these cells to staining with metal-tagged antibodies and looked at them on the site of mass cytometer. And in terms of expandability, these cells, let's do this. These cells are over 90% FOXP3 positive. And what we've done is really been able to identify actually 3 different Treg subpopulations in the liver of these children. So just as a quick reminder, the 4 groups of patients that we look at are children, liver transplanted children with alloimmune hepatitis, liver transplanted children who have normal graft function and no perturbation of their liver. We also have liver transplanted children who have acute rejection as a control. And then we also study non-transplanted patients with autoimmune hepatitis. So a lot of the data I'm going to be showing you is in these 4 groups of children. Now, what we've been able to show, as I said, was we've identified in in the liver 3 specific Treg subpopulations. We've actually seen this also in the blood, and I'll show you some data from the blood that have a distinct phenotype. So the first subpopulation is really this adenosine cyclic AMP-mediated suppressive type phenotype with double positivity of CD73 and 39 within the FOXP3, CD127 low Tregs. We see this in the liver, but we also see this in the Tregs of the blood, Tregs in circulating blood as well of these patients. This is just showing us again NRP1 of the Treg cells in the blood. We also see a second subpopulation of Tregs that have this activated apoptosis-prone phenotype with an abundance of HLA-DR and CD95 within resting or naive Tregs. And this is the same both in the liver, but also in the peripheral blood as well. And the third Treg subpopulation we're seeing is really this antigen experience, a Treg subpopulation with an antigen experience phenotype with an abundance of HLA-DR and CD73 within the CCR4 positive effector Tregs. We see this in the liver, in addition to seeing this in the blood. And so one of the questions, obviously putting on my clinician hat is, well, what do these different Treg subpopulations mean in terms of disease outcome and how these patients are doing? And so using, actually before I get to that, in terms of the stability of these Tregs, we've actually tried to study the, looked at methylation at the FOXP3 TSDR, and the FOXP3 TSDR CpG sites in these expanded Tregs range between 64 to 99% unmethylated, suggesting some variable demethylation and variable stability of these Tregs. And so back to what I was saying is trying to understand what these different Treg subpopulations mean in terms of disease outcome of these patients. What we've done is we've run a Spearman correlation. And what we've used as a surrogate of disease activity has been the liver enzymes or serum aminotransferase in the blood, the levels of those. And the first thing that we see really truly is the Treg subpopulation that's suppressive and has an antigen experienced phenotype. They seem to strongly correlate with one another with an R of 0.94. Additionally, these two Tregs, so the Treg subpopulation with the suppressive and the antigen experienced phenotype, are negatively correlated with serum aminotransferase levels. So this is kind of like a surrogate for disease activity. Now, this is obviously still very preliminary, and we still need to validate this in a larger cohort of patients. And so I think I'd just like to just end here that we have reported and shown the involvement of ERVs in the pathogenesis of alloimmune hepatitis. This is really ER stress mediated through ATF6 alpha. And we've shown that this contributes to Treg plasticity, but very, very preliminary work has identified these 3 subpopulations of Tregs in the liver as well as in the blood. And the Tregs with a suppressive and antigen experienced phenotype appear to be negatively correlated with disease activity in these patients. But again, this is something that we still need to validate in a large number of patients. And with that, I think I'll just stop here and be happy to take questions. Thank you very much.
That was a fascinating talk. Thank you very much, especially how you come from this observation that you observe autoimmune hepatitis in immunosuppressed patients to this discovery. This question may be tangential, but in our patients with metabolism or metabolic dysfunction associated steatotic liver disease. The involvement of ER stress and the unfolded protein response has obviously also been invoked. And I'm fascinated by this observation and the mechanism how ERVs can contribute to that same mechanism. Do you think the involvement of ERVs can also be contemplated in other forms of inflammatory liver disease, where maybe we are all blinded by the high frequency of fatty liver in general, but what we can't explain is why some patients obviously get aggressive inflammation and others just have steatosis, and could this have be part of that mechanism too?
Well, I think it's a possibility. Why do I say that? Because we all have ERVs forming maybe 8% of the human genome. So the question is whether or not those ERVs are activated in those patients. I think, funny enough, a couple of weeks ago, someone also brought this point up to me and asked, is this something that we've looked at? But we haven't looked at it specifically ourselves because it's just not been an area of focus for us. But I think it is possible. Whether it'll be interesting to see whether ERVs are also activated in patients who have aggressive disease compared to those who do not have aggressive disease.
Are the ERVs present in the liver, which is being transplanted? And if so, does it serve any useful purpose to know that before transplant is actually utilized, and perhaps even screen whether a transplant is suitable?
Well, I think that would be very difficult to try and exclude the presence of ERVs because we already have an organ shortage to begin with. And so we really can't be very fussy about, we can't be too fussy about the organs that we get. And you really wouldn't be able to get, even if you were to say, you know what, let's look for ERVs in donors before we transplant them, you wouldn't have the results back on time for you to use an organ anyway. Besides, we really have a problem with organ shortage at this time. So really, truly, I think our focus is more right now, certainly in pediatric liver transplantation on long-term outcomes. So our focus really would be to try and prevent this development of alloimmune hepatitis as best we can, or at least treat it better than we're doing right now. But I don't think trying to stop those organs being used is going to be a route that we're going to be we're going to have that leeway to do, just because we really need more organs, not less organs right now.
I have a question that's related to wins over here. So I think that, these are very interesting observations, and I'm wondering how you think about causation. I think you very nicely showed using like a related disease control in the form of acute rejection, that the ERVs do seem to be specific to the alloimmune hepatitis. But I think there are just like inherent limitations of using end-stage tissue to understand causation, because, for example, the Tregs, this could be a suppressive population that arises in response to some other underlying etiology. So I'm wondering how you think about causation and whether you've thought about doing like longitudinal immunophenotyping of serum or of the blood to get a better sense of what are the immune cell populations that might be driving this in the first place.
So you mean looking really before development of disease? Is
exactly.
That? Yeah. So that's going to be very, very expensive. Because if you look at the frequency, even when we looked in 5 centers and had a denominator of about 1,800 liver transplanted children, we came up with just 31 children. So that's going to be a lot of children that you're running, longitudinal testing on, looking at their regulatory T cells. So that's kind of very, very expensive. So I don't think we have that luxury of doing that. But your point is well taken. But I think it's a bit challenging for us to do that because of the expense involved.
But if you break the screen for patients who are at risk of developing this, for example, by looking at like pre-transplant presence of herbs, that would give you a population
Yeah.
that would have a higher prevalence.
Yeah. That's a good point. If we looked at that, yeah, that's a good point. That's a good point. I didn't think of that, but that's a good point. Yes, thank you.
Similar question, I guess, in terms of predictive markers, perhaps. Would maybe very specifically, when you have patients that need a second liver transplant, do they also have a higher chance of recurrence of the alloimmune hepatitis? And secondly, are there comorbidities of other allo or autoimmune that do they continue if there's a second transplant? I don't know if you have enough data to have.
Yeah, so I don't have data that shows, not just for me, but other centers that shows once you've developed alloimmune hepatitis, you also, and you get retransplanted, you get alloimmune hepatitis again. The couple of patients that we follow, and so far, who needed to be retransplanted, and so far they don't have any indication of that. But that's, just a couple of patients in my experience. But there isn't anything that's reported out there in the literature. And your other question was, I'm sorry, whether they have other comorbidities, autoimmune-like comorbidities. No, they don't. And this is something that we've always looked for. So we're not seeing thyroid disease or diabetes or, joint disease or anything like that that has an immune underpinning in these patients. This is just the only thing we see in their transplanted allograft. That's it.
Yes, great talk. So I have a question about, so do you know in which types of cells these herbs are activated? Are they activating all types of cells or in some specific type of cells?
So that's what I'm really hoping that work with the Germain Lab is going to help us answer that question, because we don't know that. We just know, when we've used the FFP that they're upregulated, but, I think it's important for us to know which cells are actually activating these herbs. So I'm hoping that our work on using spatial, high-dimensional multiplex staining would be able to help us answer that question.
Thank you, Dr. Ekong.
Thank you so much.
Okay, great.
Is that
Yeah.
you clicking now? Is that
I think that's him clicking, but okay, great. So I'll just jump right into just this brief introduction, but the title of this microtalk is Proteolethargy as a Pathogenic Mechanism in Chronic Disease. And as we've heard, chronic diseases like type 2 diabetes, fatty liver, and other inflammatory disorders affect millions of people worldwide. Unlike monogenic diseases, where there's a clear cause and effect relationship between gene mutations and the underlying molecular pathology, many chronic diseases lack a single clear cause, and the underlying mechanisms and factors that contribute to disease can be very complex. Further complicating this picture is that diverse cellular processes like metabolism, signaling, and gene regulation become dysfunctional in many of these chronic diseases. And so these challenges really limit new therapeutic hypotheses and the development of new treatments. So using a combination of live cell imaging approaches and cellular models of liver disease, we have found, my team and I have found, that the mobility of a diverse set of proteins slowed down in the process of disease pathogenesis. We've seen that this slowdown of proteins, which we term proteolethargy, is the outcome of a high ROS environment that consequently impacts surface exposed cysteines. And furthermore, we've seen that diverse pathogenic factors ranging from different dietary factors like hyperglycemia, dyslipidemia, and other factors like genotoxic stress, chronic inflammation, and other toxins also contribute to this observation of proteolethargy. I'm trying to change the slide, but I can just go on to, so on the next slide, it just is talking about how proteolethargy, we've seen that this is caused by pathogenic signaling that causes an increase in ROS and can also be phenocopied by oxidizing agents like hydrogen peroxide. Furthermore, proteolethargy can be reversed or prevented by treatment with antioxidants or cysteine capping agents. And so something that we're curious about is whether metformin, which is taken by millions of people to alleviate symptoms of diabetes and insulin resistance, if this is working by reversing proteolethargy, in large part because metformin is a complex 1 inhibitor of the electron transport chain and is thought to work by limiting oxidative stress. And so you can come check out my poster, it's poster 12. And with that, I would really like to thank the organizers of this meeting, as well as my amazing team who have contributed to this work. Thank you, and come check out my poster.
Thank you, Shannon, for a great talk. Next, I'd like to welcome Alex Ledet, who's an MD PhD candidate in the Arias and Cuervo labs, to give his talk.
Okay. Cool, great. Good morning. I'd like to thank the organizers so much for giving me the opportunity to speak. And I'll be telling you all about my project, which concerns, as you can see, the role of chaperone-mediated autophagy in MASLD. So chaperone-mediated autophagy, or CMA for short, selectively degrades proteins that bear a specific amino acid targeting sequence. These proteins are recognized by the HSC70 chaperone, which delivers them to LAMP2A. And once they're bound to LAMP2A, they can cross the lysosomal membrane and undergo degradation there. We know that CMA activity fails in conditions like aging and also in response to persistent nutritional challenges. And so for my project, I'm particularly interested in understanding the role of CMA malfunction in the context of MASLD and its more progressive forms like MASH. So in order to study these diseases in the lab, we utilize animal models. For the sake of time, I'll just tell you that we use chemical plus diet models. And so our first question, as you can see, was whether CMA activity changes during MASH development. And so to that end, we use this mouse model in the lab, which expresses an endogenous reporter of CMA activity, which we can measure as fluorescent puncta. And what we see is that CMA activity changes in a cell type specific manner during disease progression. And we see that these changes in hepatocyte CMA activity are driven partially at the transcriptional level when we consider our expression of components in the CMA network and our predictive CMA score. And using publicly available data, we see that humans in the MASH stage actually show these same changes. And so we think this may be important as a part of the protective response to this challenge. And so if that's the case, we need to know what happens when CMA malfunctions, as it does during aging in humans, And so to that end, we use the same model in mice that are deficient in CMA activity, either systemically or only in hepatocytes. And for the genotypes, they'll be indicated by the colors on the bar graphs. And what we see is that our systemically CMA deficient animals show significant increases in the liver index, while our hepatocyte specific CMA deficient animals show significant increases in the number of lipid droplets per cell. When we looked at the inflammatory component, which is an important part of this pathology, we see that systemically CMA deficient animals show significant increases in macrophage infiltration, and that when we characterize this more comprehensively using flow cytometry, blockage of CMA either in hepatocytes or in the whole body markedly changes the immune system. And we've confirmed our initial report of increased macrophages, but also we're really interested in this massive expansion of neutrophils. And so as you know, or maybe I'll mention, these cells have key roles in promoting inflammation during this disease. And we actually have selective small molecule CMA activators. So part of the ongoing work now is to see if we can use these molecules as a therapeutic target. And with that, I will thank you for your time. And I would love to take any questions at my poster, which is number 9, I think.
Thank you, Alex. Next, I'd like to invite Dilnar Mahmut up. Dilnar is a PhD candidate in the Gouon-Evans Lab at the Boston University School of Medicine.
Thank you for the organizers for inviting me to speak. Today I'll be talking to you about my project on human iPSC-derived endothelial cell therapy to repair the liver vascular network and alleviate liver diseases. So we know from a growing body of literature that after injury, such as partial hepatectomy, hepatocytes release VEGF-A, which in turn activate liver sinusoidal endothelial cells through their KDR receptor to release supportive factors, mitogens such as hepatocyte growth factor and several Wnt to stimulate hepatocyte proliferation. And so while we can all agree that the desirable cell type to transplant in liver diseases is hepatocytes, one limitation is that there's poor maturation of human iPSC-derived hepatocytes, and this prevents their preclinical benefit to rapidly replenish the hepatocytes during acute liver injury. So we propose as an alternative strategy is to transplant supportive endothelial cells. So this leads to my hypothesis that human iPSC-derived IECs therapy will not only restore the damaged liver vasculature, but can also alleviate liver damage by inducing proliferation of the remaining mature hepatocytes through the host hepatocyte and IEC niche interactions, resulting in secretion of the supportive endothelial factors. So first, to do so, we generate these IECs using a protocol developed by Alex Holt, who is a collaborator of ours at the CREM. And he's taken key aspects of these previously published studies, and we can generate pure endothelial cells in culture. And we can see that they express CD31, CD144 through flow cytometry at day 11. And when we look at immunostaining, we can see that they also express KDR and as well as live-1. So to test IEC engraftment and whether KDR activation can promote their engraftment, we transplanted 1 million IECs intrasplenically into NSG mice with a monocrotaline injury, and we delivered VEGF-A mRNA LNP at the time of transplantation and once a week for about 3 weeks. And our lab has previously shown that mRNA LNPs are a very robust tool to express proteins in the liver and that they are transient. So we can actually track the fate of the cells since my starting cell line has a luciferase construct. And so we can track the fate of them using IVUS imaging. And from our luciferase data, we can see that VEGF-A promotion really expresses persistent and robust expression here in our treatment group. And when we look closer at the cross-sections of these livers, the mice treated with VEGF-A mRNA LNP have larger clusters of transplanted cells. And here in a close-up, I can detect the cells using a human CD31 antibody. And when we look to see whether they are in the vasculature and the sinusoidal network, we can see that they do line up where we have mouse KDR expression. So currently, we're actually testing to see if IEC and transplantation can alleviate liver diseases and specifically help accelerate liver repair. I only had 2 slides, so I don't have an acknowledgement, but thank you to everyone listening, and my poster is 13. Thank you.
Hello, everyone. Welcome back for session 2. I'm Caitlin Bowman, an assistant professor of biology at Williams College. We're delighted to welcome you back.
Hi, everyone. I'm Lucia. I'm a graduate student in Sangeeta's lab. And first of all, the first speaker is Dr. Ann Carpenter. Dr. Carpenter is an institute scientist at the Broad. Her research group develops algorithms and strategies for large-scale experiments involving images. The team's open-source cell profiler software is used by thousands of biologists worldwide, and their cell painting assay has been adopted throughout the pharma industry to accelerate drug discovery. She leads industry-wide consortia such as Jump Cell Painting and Oasis. Her PhD is in cell biology from the University of Illinois, Urbana-Champaign. Her postdoc is in high-throughput image analysis, was at the Whitehead Institute for Biomedical Research and MIT CSAIL. Dr. Carpenter has been named an NSF Career Awardee, an NIH awardee, and is listed in Deep Knowledge Analytics Top 100 AI Leaders in Drug Discovery and Advanced Healthcare. Dr. Carpenter, the floor is yours.
Thank you. I've got this.
Hi, everyone. Oh, can we get the front-facing monitor showing? Maybe not, but I'll keep turning around. So hello, everyone. I'm really excited to be here. I'm not a liver researcher, but because I've collaborated for 20 years with people all across Boston, all around the world, eventually you accumulate enough liver projects to put together into a talk like this. So I'm happy to tell you how the world has been changing with respect to microscopy images and how they've turned quite quantitative and how they are now being used in drug discovery. And I'll be using a number of different liver examples in the face of that. So there's been quite a revolution in how we use images. In the old days, by which I mean not really that long ago, people have been using microscopy images for screening to identify new drugs. And more recently, we have begun using all the information in images in more of a profiling way, and that's called image-based profiling. So the talk's divided into two chunks, and I'll give some examples of each in case all of these words are unfamiliar to you. I'm living in the world of high-throughput cell-based experiments where we're trying to rapidly discover the causes and cures for various disorders. Okay, so just to get us all on the same page, when we're thinking about drug discovery, it'd be very nice if data could help us along in this pathway. And so you, I'm sure, have seen headlines about machine learning and artificial intelligence are going to revolutionize drug discovery. A healthy human. This is what clinical trials are, but first of all, we fail 90% of the time. Even if we spend decades trying to design a chemical that does a good job, our ability to make these predictions is pretty faulty at best. But we can't scale this up. Scaling it up is not going to help. We already failed quite a lot. And so we try to make some approximations in the lab. So most of the experiments I'll describe today use cells as proxies for humans. So instead of testing diseased patients where we do such a bad job, can we come up with a system in a dish that represents the disease in such a way that we can then test lots and lots of chemicals against those disease cells and end up with healthy cells? So here's one example assay where we have cells infected with tuberculosis, and our goal is to find a drug that can make the tuberculosis bacteria go away, but the human cells are happy and healthy, just as one very simple example. So my lab is helping people to extract the information they need from images to carry out assays like that at large scale. And these experiments usually involve testing lots and lots of samples in these 384 well plates. So each little teeny well on this plate has cells growing in it, and then you image it through the bottom to identify, through the clear bottom of the plate to identify the cells that are growing there. We use software for what's called segmentation. The image analysis that goes into this process involves identifying the outlines of all the cells. And my career got started in this space because there existed no good software to do this. And so I started writing Cell Profiler during my postdoc and ended up, now it's worldwide. Usage is in the pharmaceutical industry and in academia as well to be able to do these kinds of high throughput experiments. It's a pretty solved problem, I would say, these days, especially with deep learning methods. It's now very straightforward to identify lots of things in images and then make all kinds of measurements. Some you would think of and some are kind of more obscure, but we're measuring things like, first of all, how many cells are present? What are their shapes and sizes? The different compartments of the cells, do they have different staining levels of whatever you've stained your cells for? Different textures, correlations, and so on. So tons of things can be measured out of these images. And that allows us to create assays that represent all kinds of different diseases in a dish and then test lots and lots of drugs or genetic perturbations in a very rapid kind of way. So as I said, it's become a relatively solved problem. There's all kinds of edge cases, all kinds of more difficult situations like, let's say, neurons growing in images can be kind of difficult to trace. Some organisms, if you're imaging a whole organism, it can be a little difficult to find particular organs you might be interested in. But if we're talking about cells growing in a monolayer on a dish, usually software does a good job of that these days. So this work has been taken over by an alum from my lab, and maybe the most important thing if you work with images is this image.sc is a free online forum where people ask tons of questions about any image analysis problem and get it solved. So over the past 2 decades, we've done dozens and dozens of these kinds of image-based high throughput experiments where we look for drugs or genetic perturbations. And here's just a couple of examples that have led to successful clinical trials. The first finding a drug that allows the cells to, myelofibrotic cells to basically instead of continuing to proliferate, they sort of differentiate and end up in this sort of static state. And that's proven to be helpful for patients with this disorder. And then another project, which actually we had nothing to do with, but they just used our software, involves leukemias and lymphomas, where in this personalized medicine experiment, researchers took tumors out of a patient, put them in a dish, tested different potential chemotherapies for each patient, and then selected the therapy based on the actual patient's cellular response. So a little bit of the wave of the future there. So I picked a couple of examples that we've done in the space of liver. Both involve Sangeeta Bhatia's lab. And in this first one, you'll see we took some human primary liver cells, and this was a student in her lab that carried out all the physical wet lab experiments here, and co-cultured them together with fibroblasts so that they'd be happy and healthy and exhibit enough of their normal properties so that we could use it as a good test case. And then our lab's job was to figure out, can we distinguish the human hepatocytes? Those are the ones marked with a little yellow star. They're these a little bit smaller, a little bit rounder, a little bit smoother than the mouse fibroblasts that are growing alongside them. And we used machine learning, which was revolutionary back in the time. This is now more than a decade old. And we were able to distinguish those two cell types so that we could simply count them up, how many hepatocytes are growing. And that way, the researcher could test lots and lots of chemicals to identify those that stimulate primary human hepatocytes to proliferate and differentiate. And the second example I'll give is, again, using human primary liver cells, this time infecting them with malaria. Those are the little red dots that you see here. And here, again, it offers the opportunity to, in a scalable way, we run the image analysis to identify and count how much malaria is present in the context of the human primary liver cells growing. And the idea here, because malaria likes to hide out in a rather dormant state within the liver, it offers the potential to really rid patients of malaria for good, if we can find drugs using this kind of an approach. Okay, so those are some examples of what is called high content screening. The name comes from high information content screening. And what it's a kind of weird phrase, but what the people mean by it is microscopy-based screening. So let's do high throughput microscopy, test lots of samples and take images of things. But high information content is a little odd when, as I just told you in the experiments I described, we're really just measuring one particular thing, like how many malaria dots are there, or how many tuberculosis dots are there, or how many hepatocytes are there. And so some can be more or less complex. Sometimes it takes machine learning to measure the thing you want to measure. But there's actually quite a lot more information in images than just what we already know we want to look for. So the second part is talking about image-based profiling, where we take images and take all the data out of those images and use that to characterize samples in a much broader way. So this is pretty important to understand the rest of the talk. It's image-based profiling is where we use images to create these signatures of our samples. The samples can be genes, they can be compounds, they can be diseases. So these could be different patient cell lines or cells treated with different drugs or so on. We've stained the cells with whatever set of stains. Here I'm showing cell painting, and I'll talk about that in a minute. Stain the cells somehow, extract all of their numerical features in a way that I just showed how we can do. So we end up with thousands of features times thousands of cells for every individual sample. And now what you can do, just even looking at this like teeny tiny chunk of real data from a real experiment, you can say, okay, well, I see some patterns there. So the individual cells, usually have a pretty consistent response to whatever the treatment was in each sample. And I could now start grouping samples together that look alike. And I can start differentiating samples that start to look different from each other. So you could imagine using this approach, for example, to separate patients that have a disorder versus patient cells that have a disorder versus healthy controls. Is there anything about the images that looks different among them? And this is what we mean when we say profiling. We're taking a rich profile of data and using it to compare samples. Okay, so there's these next 3 slides, I admit, are a little bit overwhelming. It's really just, the point here is to just give you a sense of the kinds of things you can do when you treat images in this way. It's very much like transcription or proteomic profiling. If you are familiar with the kinds of things you can do with those sorts of profiles, it's just a heck of a lot cheaper to use images as the readout. So we can do experiments that are tens or hundreds of times larger for the same cost. So for example, we can group genes based on their functional similarity. So if I knock out 2 genes and they have the same impact on the microscopy images of the cells, that gives me a hint that those 2 genes have similar functions. That's the basic concept. So we've made tons of data in this space and demonstrated that principle. We can even go so far as to group alleles by their impact. So here, if we overexpress a protein with and without a human mutation, we can compare, does that mutation impact the morphology of the cell in any particular way? And there's examples of that. We can group chemical compounds. So if we treat samples with different drugs, 2 drugs that have a similar mechanism of action will look like each other in terms of the literal microscopy images, but also in terms of the image-based profiles. So we can quantitatively group thousands and thousands of compounds. This has been pretty well incorporated in a lot of pharmaceutical companies today, where they'll try to figure out the mechanism of a compound that they're interested in based on its image-based profile. We can also use it to identify disease phenotypes. So again, I won't go into any details here, but these are different examples where normally researchers would use very fancy antibodies, sometimes expensive equipment, to try to identify different cell types based on expressing particular markers. And it's turned out that there's so much information in images, you don't even need the antibodies, you don't even need the biomarkers that people thought were necessary to identify a particular cell type. Instead, you can just take bright field images, these grayish examples are examples of bright field images, and just extract all that information and identify, for example, leukemic versus non-leukemic cells, or different white blood cell types that even human researchers can't distinguish by eye, but the computer is able to distinguish. And then lastly, image profiles can make all kinds of different predictions. We can identify human genetic variants that mediate particular types of cell morphology. We can predict whether a compound is going to be active. So we can screen hundreds of thousands of compounds to identify in just a computational query which one is likely to have the impact that I want on different assays. And we can identify potential chemical regulators of pathways as well. And so I want to zoom in on this making predictions, because that's where it gets to another of our interesting liver related projects recently. So can we predict chemical properties and toxicity from images? So the idea here is, can a pharma company or an academic institution test all of their compounds once in an image-based assay, capture those images, collect the profiles, and save them in a database forever and ever. And then whenever a researcher comes and says, oh, okay, I've developed a very fancy assay for this, or I have some compounds that I know produce mitotoxicity or cardiotoxicity. If I have a collection of known correct answers and known incorrect answers, can I train the computer to look at those images and predict the outcome of my assay? So in other words, the pharma company does one assay one time with imaging, and then can predict the results of maybe 100 assays going forward that are predictable. Not every assay is predictable using just one particular image assay and one particular cell type, of course. But this works often enough that it's worth giving a try, because it saves tremendous amounts of time and cost to be able to predict assays rather than physically running them. So there's a bit of proof of principle from these predicting these different types of toxicity, but what I want to get to is our mission to be able to predict hepatotoxicity simply by snapping a picture of cells that have been treated with a given drug. And not just drugs, actually. This consortium is not just pharmaceutical companies, but also agrochemical and consumer chemicals as well, because they have every bit as much interest in making safe compounds that do not affect the liver. So our goal in this consortium that we just launched last year involves a couple dozen different companies, where our goal is to test cell painting, the image-based assay that I've been talking about. And I can give more detail if anybody cares about the precise details of it. But that image-based assay compared to transcriptomics and proteomics can we combine all of that data to make good predictions about a compound? The idea here especially is to avoid using animal models, partly for ethical reasons, but partly for cost reasons, and partly because they just don't predict hepatotoxicity very well. So for all those reasons, we would love to find some kind of human in vitro cell-based assay that would allow us to predict compounds, even better if we can do it at a very large scale. And so this is the OASIS Consortium, and this slide gives you a sense of what the organization and plans are. We've taken more than 1,000 compounds from the public domain where there is some, the requirement here for a compound to come into our pipeline is that it has some kind of in vivo data in the world. It can be rat data, it can be human data, but we want to have some kind of sense of what this does to the liver in an actual living organism. So that limits it pretty, I mean, this is essentially the universe of compounds that exist in the world, a bit over 1,000. And we're going to include some from industry partners as well. And what we will do then is use a number of different cell model systems. These range from being super cheap to super expensive. They range from easy to run on 1 million compounds in a pharma environment to you probably don't want to run more than 10 or 20 at the most expensive end of the spectrum. But we'll be doing a range of these, especially in collaboration with some of our technology partner companies. And then we'll, for each of those, we'll be doing different types of readouts. So these are the different assays we'll be using: images, as I described, transcriptomics, and proteomics. These are the more high throughput versions of the mRNA and protein profiling. And so with all that data, we will combine it, try to figure out if there are any of those combinations of cell models and readouts that predict different types of hepatotoxicity. This is not a simple machine learning problem where, you know, we have a pile of toxic and a pile of non-toxic compounds. As I'm sure everyone's aware, the concentration is what makes something toxic. And so that's all the data analysis team is working on that. And in the end, we'll release, of course, everything publicly, and hope to develop a community of researchers within all of these companies who are able to use these technologies to accelerate drug discovery and agrochemical and other chemical discovery. So we just have our first little bit of data from this consortium, and that's thanks to a collaboration with Axiom, one of the tech partners, who was able to run 3 assays side by side. And amazingly, they were able to use primary human hepatocytes. That's not cheap or easy, but important for looking at, you know, the realistic response of liver cells to these different compounds. They had more than 1,000 compounds in this dose response curve. And these readouts included the cell painting assay, which is the imaging, as well as 2 biochemical assays. The MTT, we won't go into detail, is sort of metabolic, and LDH is an assay that sort of tests whether the cell has exploded in dramatic fashion, just long story short. And so here's how it looks. What we're interested in finding here are which of these technologies allow us to detect toxicity to the liver at the lowest possible dose. And so the y-axis here shows the chemical dose where you start to see a chemical effect. And each dot is a different chemical that we've tested in the experiment. So if we start at the LDH example, most compounds, you have to get really high doses before you start to see an impact. You start to see the cells exploding. And then there's a few trickly examples of compounds where you can use a much lower dose and the cells respond. If we just count the cells in the images, that's the cell count, it's a little bit better. We get a little bit more sensitive. MTT as an assay is a little bit more sensitive. It's not waiting for the cells to explode. It's just testing, is there something metabolically off with these cells? So you get a little bit more sensitive. But if we use the images, as you can see, we have a much greater sensitivity for most of the compounds. We can tell something's going on with the liver cells in a dish. At these much lower doses. So it was pretty exciting early evidence from this consortium. And as you can see, the numerical quantification there, more than half of these toxic compounds could be detected in a single assay, which is pretty exciting, especially if you can compare that. If we can reduce animal testing by 50%, it would have really dramatic, not just dramatic impact on the use of animals and the cost of using those animals. But what it allows us to do is, this is cell painting such an easy assay to do. You can run it very early in the drug discovery process, which means you're making decisions based on liver toxicity early in the process, rather than waiting till you've got a drug that you're really excited about, you really think it's going to cure your disorder, and then you're just kind of hoping that it's not going to be hepatotoxic. We can incorporate those findings much earlier in the drug discovery pipeline. So I hope you enjoyed this little super brief overview of how images are being used at different points in drug discovery. There's a PDF of these slides are available online. Everything I talked about is public today, so you're welcome to take pictures or anything. This gives you all kinds of different references for review articles and conferences that work in this field, all kinds of other types of applications of this that I didn't have a chance to talk about today. And with that, I thank you very much for your attention.
Thank you so much, Dr. Carpenter. We have time for 1 or 2 questions, and we have plenty of time to chat later.
Just, yeah.
All right, great. Thank you for the beautiful talk. It's super exciting to see how we're starting to use this high content imaging more and more frequently in the lab. One of my questions was related to the sensitivity data you showed at the end there, that the imaging is picking up changes earlier on or at lower doses. And something I was wondering about is, do we have any insights into knowing whether the changes it's picking up are biologically meaningful?
Yeah. I love the question. These results are hot off the presses as of 2 weeks ago. So then one of the next questions is to dig into cases where the other assays completely miss the toxicity. What are the particular features that we're seeing in the images that help us understand what the cells are responding to or what pathways are activated? One really cool aspect of that is to say, okay, we see a particular profile. Are there particular features that are prominent? We have other data sets in other cell types and elsewhere where we can say, have we ever seen any other gene perturbations or chemical perturbations that have yielded that? And that can give us even more insight into maybe what pathways are being disrupted and so on. So especially in the field of toxicology, it seems so important to map different layers. You know, when we say this is toxic, what do we mean? There's, you know, the compound binds a protein that has an effect, that has a secondary effect and a tertiary effect, and eventually you get the liver being sad. And so looking at each of those individual steps along the pathway, we're really excited to work out mechanisms. Thank you for that question.
Thank you so much. Okay, it looks like we don't have any other questions, and we'll go to the next speaker, which is Fernando Camargo. Dr. Camargo joined the Children's Hospital and the Stem Cell and Regenerative Biology Department at Harvard University in 2009. Dr. Camargo was named a 2009 V Foundation Scholar and is the recipient of the NIH Director's New Innovator Award. Most recently, he has received the Wilson Prize for Creative Promise in Biomedical Science and the Dr. Susan Lim Award for Outstanding Young Investigator. In February 2022, Dr. Camargo was named the first Regenerative Biology Endowed Chair in recognition of his scientific contributions and strong leadership. Dr. Camargo, the floor is yours.
Thank you. Thank you, everybody. Can you guys hear me? Yes. Okay, otherwise I'll scream. All right, so thank you so much for the invitation. Pleasure to be here. It's my first time here. Hopefully I get invited again. There we go. My lab works on a lot of things, hematopoiesis mostly, but we do have some work in the liver as well. And what I'm gonna show you today is some, I guess, tool application of, you know, some techniques that we've been developing to study hematopoiesis and now can be applied to solid tissues. This is the first time that I ever show this outside of Children's Hospital, so we'd love to get your feedback. All right, my lab has been passionate. You know, we have a very strong passion about studying ancestry, simply because we think it's important for many reasons to try to understand where cells come from. Right, so just like we come from a home and a lineage, an ancestry, we think that where you come from, right, who your ancestor is, will have an important value in deciding who you are eventually and how you do the job that you do. So we think there's functional consequences for this. And we want to start by identifying cell origins, cell ancestries. And in addition to that, whenever we started the lab, this was more than 15 years ago, we really thought that in order to study this, right, you would have to develop tools that would also speak to the individuality of the cells. This is before the single cell revolution. Now I think everybody understands the power of looking at individual cells and not a pool of them. Okay, so we are developmental biologists in the lab, stem cell biologists. And one of the techniques that, for the ones of you that have worked with some model organisms that has been particularly powerful, is lineage tracing. And in the mouse, historically, lineage tracing is performed using these Cre recombinases, right, that some of us have played around with. And then they allow for the activation of a reporter in a particular cell type that expresses the promoter that drives that Cre enzyme, okay? So, and your readout can be single color. You can see these beautiful colors here. The readout can also be multicolor, right? And there's been some very exciting work in multiple tissues that look at the clonality, meaning if you find a single color, right? Especially in a tissue that has physical boundaries, then you can make certain assessments about individual behavior. Okay, so this has been sort of the main, I would say, workhorse to study lineage, at least in the mouse and other model organisms. But there are clearly some limitations to using Cre and the other recombinases that are out there. First of all, for the most part, the cell tracing again is at a population level, right? If you have a promoter, say albumin Cre, right, that traces all the cells that express albumin. If you want to look at, for instance, clonality again, right? So this, even if you were to have that multicolor reporter, if your cells actually mix and migrate, right? So you will never be able to tell whether the cells that have the same color are actually clonal or not. And this is particularly the case for the hematopoietic system, right? Where all the progeny goes into the pool of blood. And this, you could argue, could be a bit biased, right? Because you have a requirement of a gene of interest to actually use as a driver for the Cre or recombinase, so on. So over the past, again, 15 years or so, my lab has been developing novel technologies to study lineage. And we have developed multiple tools that I guess can be defined in the umbrella of cellular barcoding. So we've generated lots of mice, and I'll tell you some about that. But I think the general idea here with these cellular barcoding tools is that you actually sort of use a different range of tricks to actually uniquely and genetically label cells, right? And that label is heritable. So you can trace, in theory, thousands, if not hundreds of thousands of cells at a time. So there's multiple applications of this. You know, you can do phylogeny, you can perform tracing in multiple contexts. You can isolate drivers of functional heterogeneity. I'll tell you a bit more about that. So as I mentioned to you, most of the work that we've done has been in the realm of hematopoiesis. And again, this describes some of the work that we've done over the past 10 years or so, where we've published multiple techniques and especially some important findings that we have done with this in the hematopoietic system. And this is all great, but you guys are not hematologists, so you don't care about this. So what I'm going to show you today is sort of the next step. Which I thought is an important step. The problem with these studies, which is not a real issue for hematopoiesis, is that in all of these studies, whenever we do the analysis of barcoded cells, we just simply take the cells, make them suspension, and run them through a single cell machine, right? I don't have to tell you, and sorry, before I go on, let me just explain you the genetics a bit of the system so you understand how this works, right? This is one of the first models that we published. It's called Carlin. And it basically uses an inducible system for Cas9 expression that then allows for barcoding at any point in the mouse development or adulthood. So the idea is that these mice have also an array of targets for sgRNAs, in this case 10, that based on the quasi sort of random indel pattern post-Cas9 activity on these target sites, you get these sort of barcodes, right? They're not completely perfect. Many times you have the same barcode if there's a common edit, but you get a diversity of deletion insertions that can be called barcodes. And in this particular model, these barcodes are actually expressed. This is the target array. The genetics don't really important. We can talk about that later. But this is an inducible model where you give doxycycline. There's editing in this target RNA and this target sequence, and that target sequence is expressed so you can detect it using your traditional single-cell RNA-seq procedures. So the first iteration was pretty good, but there are some issues. The overall diversity was not that high. So if you wanted to say barcode a whole mouse or barcode hundreds of thousands of cells, you don't have that diversity. And especially the barcode capture at the single cell level was actually pretty low. So just last year, we published the second iteration called Darling with a couple of different features, meaning that the Cas9 was now replaced by a Cas9 TDT, which biases the edits towards insertions, right? So you don't have full deletions of that barcode, which would diminish diversity. The arrays, now we have 3 arrays as opposed to 1 for the barcoding. The arrays are expressed at higher levels. The editing is better. And overall, if you just focus on this, and if you're interested in doing these experiments, these mice are available now through JAX. And I would say 70% in a typical experiments of all the cells that you actually run through a single cell machine, I would actually give you important information about lineage. Okay, as I was mentioning to you, I didn't do anything. There we go. The problem with the experiments in the hematopoietic side is that we do not have that special information, and we didn't really care much. But for some tissues such as the liver, we do need that information. And I don't have to convince you that it's important to know this, all the important information that you could get from being able to know where your cells are, who they interact with, and so forth. Okay. So what I'm going to tell you about today is some of our first examples, advances in trying to actually do this barcode readout completely in situ. Okay. And this is work done by Joyce Gia, who has a PhD from UT Southwestern. She's worked in the liver there as well. So she was very excited to actually try to get this going. And the idea here is to actually use some of the sort of novel spatial transcriptomic techniques that have been discovered, some of them in this building, and apply to our sort of latest generation barcoding mouse. And that's the data that I'm going to show you today. And so to make a sort of a long story short, Joyce really did an amazing work trying to optimize this. She's developed SpaceSeq, which is basically utilizing this SliceSeq technology that Faye developed here at the Broad, which, as you may know, simply uses beads, physically positioned beads as sort of pixels for gene expression, where they can also, where you can then match up your tissue and then get these, very cool images of gene expression in the context of space. So she's modified the protocol to actually being able to detect now all the barcode arrays expressing in these Darling mice. Okay, so we can detect gene expression, overall gene expression, and we can also detect the barcodes that are expressed. And this works quite well. Otherwise, I wouldn't be here. And just a quick sort of image before I go into the liver, because the brain also is sort of the tissue that works the best in SliceSeq for whatever reason. This is a mouse rhesus that was labeled in the neonatal stage and follow up all the way to, I think, 1 year of age. And you can see every color here represents a different clone. And in this case, every sort of dot quasi cell, not exactly, but you can clearly see now these beautiful colors that you can get and different patterns of clonal expansion. In this case, physically constrained clonal expansion that you can get from looking at some of these images. Okay. All right, so that's this work in the liver. And the first experiment that Joyce did to try to sort of validate this even in another way was to use a liver cancer model. This is a transposon-based model with NRAS activation and knockdown of p53, where you get lots of tumors, right? And in theory, the tumors here should actually be arising from a single cell. So this could be also a good way to validate this. And this is just, for instance, one example of one of the slices here. And you can see just by histology, maybe 4 different tumors. Using gene expression provided by the SliceSeq, you can actually look at NRAS expression, which is the oncogene given by the transposon. Maybe you have 4 different tumors or 4 different nodules, right? First question is, are these clonal or not? So then we actually looked at the barcodes that we could capture from those sites. And the answer is yes. completely, right? So this can tell you this is, every dot here represents a barcode, and you can clearly see that these tumors are completely clonal. Again, suggesting this actually works well, and it can in fact detect single cell derived growths. The other interesting thing that Joyce did was to actually look at the clones in non-tumor cells, so parenchymal cells. And remarkably, she observed that in some places in the tumors, she observed this massive clonal amplification of the hepatocyte stellate cells / myofibroblasts, which was very interesting. There are other sites. I'm only showing you here the clones that actually expand. There's many other cells that we could capture that do not expand, right? So this is quite interesting. And we only saw this in the myofibroblasts, not in the immune cells or other non-parenchymal cells that we looked at. And with this, since we have the transcriptomic data from the cells, then we can actually group these myofibroblasts in clonally expanded versus clonally not, right? And if Joyce does that, we get a nice signature of genes that are actually and exclusively enriched in the myofibroblasts that expand. Could potentially mean, you know, a signature for potentially intervening in the context of fibrosis or stromal activation in cancer. And given that this is sort of spatially restricted, you can see these clonal expansions are only in certain parts of the tumor, then Joyce decided to look also at the gene expression in the tumor cells surrounding those clonally expanded myofibroblasts, okay? So for instance, in the left, I'm showing you sort of the pattern of the clonally expanded HSCs in orange and the non-clonally expanded HSCs in blue. And what Joyce did actually then, she measured the expression of genes in the tumor cells and compared the tumor cells that were nearby non-clonally expanded versus cells that were nearby clonally expanded. And as you can clearly see, there's also a very different sort of set of genes that are expressed that she's currently testing now in terms of functionality. And not only that, with that gene expression differential, she then is able to actually look in not only these tumors, but other tumors and try to figure out, for instance, these domains of tumors that typically are associated with non-proliferating or clonally expanding hepatic stellate cells, okay? So it works, and based on the extra information of the transcriptomic side, you're able to make some important sort of discoveries as far as genes that could potentially play a role. Clearly, a functional assessment is necessary to actually look at this. Okay, so the next question is, can we actually use this space-seq technology to provide some insight into liver development? And Wolfram, I hope you like this. This is sort of a general schematic of liver development. And the stage that I'm going to sort of try to focus on is this one here, when you actually have the liver bud, right, full of hepatoblasts. And there's very little data in the literature about what happens to the cells as, you know, the liver increases in mass and the liver becomes lobated. So to put it clearly, right, to what extent these hepatoblasts expand and to what extent those hepatoblasts migrate. And again, this is not something you could do with sort of a rainbow type of approach because there's many of these, and some of these might actually, if you have the same color, they might actually intermix with each other. So in the following experiments, we perform induction, so barcoding the mice either at this stage around E10, around this stage around E12, and a bit later, okay? And then the readouts are always done in the adult postnatally. Okay, so for instance, if the induction in this case is done at E15.5, the data E12 is exactly the same, so we use them interchangeably. And this is one of the actual outcomes of the SLISIC data. And Joyce has developed a very nice sort of algorithm to actually try to recreate the lobules in the liver based on the expression of periportal and pericentral genes, right? So we can virtually generate these lobules so we can actually have a better sort of view of what happens to the clonal expansion. So if you label again at 15.5, and then you try to match some clones to this slide, this is what you get, okay? And again, each color here represents an individual clone, and you can see clonal expansions in the liver nicely. But for the most part, they are completely anatomically and physically restricted, okay? Which, I thought it was cool, maybe not that surprising, but I thought it was cool. But I think the remarkable part was whenever you actually labeled at E10. And if you label at E10, you see this very, very, very cool pattern. And you see, observe two things. Number one, the clones are much larger, obviously, right? Again, each color is a different clone. And your clones have actually expanded quite a bit more. And you have this pattern of sort of expansion, right? Migration, rather, right? And you have this subclonal expansion that occurs in that period of time. Okay, and this is one clone, but you can clearly see that there's actually expansion. And the other thing that you can actually see, if you, for instance, look at individual liver lobes, right, you can see that even by inducing an E10, most of your liver lobes, lobules rather, are actually non-clonal, okay? So this to us demonstrated, right, that there is probably a short window of time developmentally, maybe between 10.5 and sort of 11.5, where your hepatocytes proliferate and can actually migrate. Okay, which was something that was unclear, at least to my understanding. And the next question was, so how far away can these hepatocytes go, these hepatoblasts go, right? And it's tough to look at this in slide C because again, you're looking at single slides. So what Joyce decided to do is to actually do bulk experiments, right? And to sort of address the question again of how widespread is this migration, right? And is the migration equal to all the lobes in the liver? And again, these are the times in which we barcoded the animals, and then in the adult, we actually physically isolated the different lobes in the mouse, the caudal, the right lobe divided in two, and the median lobe and the left lobe. Okay, and then simply isolated RNA from those lobes, and instead of doing single cell, in this case, we just simply PCR the barcodes and see what barcodes are there. And then we actually looked at ancestry, right? Which lobes share ancestry. And I guess you have two options. In one of them, all the clones will be everywhere, right? And the other side of the possibilities will be like the clones are very restricted in what lobes they actually participate during development. And the answer is here. Okay, so first of all, if you label starting at E12 and onwards, and this is looking at clonal overlaps, okay? So the degree of ancestry similarity, basically. So if you label 12 or onwards, there's actually no migration anymore, as mentioned to you, right? So there's actually no shared ancestry. Interestingly, if you label at E10, you actually have sort of two main lineages, right, in terms of the lobation of the liver. You have one lineage which is suspected, or this is the median lobe, but the median lobe seems to develop together with the left lobe. Whereas on the other hand, you have the caudal lobe and you have the right lobe that seem to be, I guess, based on this time of labeling, coming from predominantly different ancestries. Okay. So with this, we basically come up to a model in which we argue that there is this limited window of proliferation of these hepatoblasts, and that this migration is not completely widespread, right? There are certain domains in which these cells can migrate, and this is how you end up with this sort of polyclonal tumor, a liver, in which there's mostly two main lobe lineages, okay? And we're now looking at whether, for instance, the ancestry, the type of progenitor that you had has an actual functional effect on the function of those hepatocytes in the adult. Okay. Okay, so I'll just summarize saying that SPACIC works quite well. It works on liver cells, and I hope you saw evidence of that. I talked to you about the conceptual observations. And the one thing I would say to end up is that this is not quite single cell resolution at this point, and that's because of the intrinsic limitation of the technique, because of the bead size that are used. And so we have some work going with StereoSeq, which actually is true single cell, which is super expensive, but I think it's going to work. So again, happy to discuss with you and sit down and tell you about how this works, and so we can do some experiments together. Thank you.
Thank you so much, Dr. Camargo. And at this point, we have a time for a few questions. I see one up there. Sorry, I could have you answer that one. Thank you.
Thank you.
Have you looked at using spacing in human malignant disease of the liver? Have you done any collaboration?
Yeah, I think the problem with obviously doing this in the human is that you need this sort of set of recombinant tools to actually do the barcoding, right? So in the mouse, again, we use this Cas9 derivative. But there are technologies now in which you can actually do clonal tracing in the human. In situ, still, I think there's work to be done, but I think, for instance, some of the mitochondrial tracing that has been published, I've seen some data that some people are actually using some of these spatial transcriptomic techniques to be able to look at clones. So this is something we're going to start looking at, but I don't think it's quite ready for prime time, but I think it's going to be doable. Yeah, very exciting.
A question here in the middle.
Hi, it's extremely interesting talk and powerful technique. If I understand correctly, so you basically did a post-trace experiment, right? You did with one time point, you label and you shut it off. But could you progressively trace the thing to see the evolution of the traces to represent the developmental process?
Yeah, that's a great question. So this is one-shot tracing, right? So in theory, we can give doxycycline at different time points to then have a secondary and tertiary tracing. And we are doing this right now. It's not ideal, right? Because some of the barcodes that you generate in the first instance are not going to be editable. But I think that there's a lot of things that you could do computationally to try to reconstruct those lineages. The Weizmann lab here at the Whitehead has generated a system that is something like that they use mostly in tumors. But again, to me, the main limitation is that ideally you would need to timestamp every single edit, right? So we're working on techniques that will be able to do that. Maybe at least 3 different edits at the time points in development where you would want them.
So secondly, can you use some optogenetic approach that you can label really things spatially? You know, you activate Cas9, for example, just with this patch.
Right. Yeah. There's a lot of ways in which you could potentially do that. We're happy to chat some more.
And we'll take one final question up front.
Really, really cool work. And also great to see how you apply your, come down from the hematopoietic heights to the lowland of the liver.
Went down for sure.
Yeah, of course. To define this period of time when lobation, seems to be defined. My question is, do you think it just happens that the cells sort of happen to clonally expand in these different regions and then lobation happens? Or do you think at this point, there's some predetermined path for them that they already know what they have to become?
Yeah, that's a fantastic question. There is some data to suggest that at this particular time, we really need to look at the sort of anatomical literature because there's a lot of work that's been done. There's a couple of papers that I think rightly so. We haven't done this. We haven't looked at livers that carefully, but there's already some lobation patterns already at 10.5 in the mouse. It is unclear what each one of those is going to do. There's some further rearrangement that occurs after that. So I think the first thing to test will be if some of these domains correspond to that initial physical pre-lobation pattern in the mouse at 10.5. I would like to think that somehow this is more your second hypothesis, that this is sort of ingrained in the cells at some point in development so they will know where to go. So that's a cool hypothesis, so we have to test it.
And just to follow up on this, from recent work in the fish, we have learned, that at the hepatoblast stage, there may be some hepatoblasts that give exclusively rise to hepatocytes, and others are truly bipotential and can give rise to cholangiocytes and hepatocytes.
Well, we can totally test this. Absolutely.
Yes. And so you showed us here hepatocyte-specific data, but you have the potential, right, to look at what becomes of hepatoblasts in both
lineages. Great question. So unfortunately, because of the resolution, it's very hard to see ductile cells in this space, but we can simply sort them. So we could certainly do that together, and I think that's totally doable. It's a great question. Yeah.
Thank you.
Thank you very much.
Thank you.
Our next speaker and final plenary speaker for this session is Dr. Kwang-Hung Chung, who is an associate professor of chemical engineering at MIT, as well as a core member of the Institute for Medical Engineering and Science. He's also an associate member of Broad Institute. He completed his PhD training under the mentorship of Dr. Hong Lu, where he developed automated and integrated microsystems for high throughput imaging, molecular behavioral phenotyping, and cell microsurgery. And then he joined as a postdoctoral fellow of the lab of Carl Deseroth at Stanford, where he invented the novel technology termed Clarity. This enables system-wide structural and molecular analysis of large-scale intact biological samples, about which we'll hear more today. Dr. Chung joined MIT in 2013 and has been leading an interdisciplinary team developing further applications of this novel technology. And his group has developed technologies that enable rapid and scalable 3D imaging and phenotyping of both animal models and human clinical samples, which we'll hear about today. He co-founded Life Canvas Technologies to commercialize these inventions, and you can learn more about that from him later today. So with that, I would also like to mention he's the recipient of several awards, including the Presidential Early Career Award for Scientists and Engineers in 2019, the NIH New Innovator Award in 2016, and the McKnight Technological Innovations and Neuroscience Award in 2016. Thank you, Dr. Chung.
Thank you for the kind introduction. Good afternoon, everyone. Can you guys hear me okay? Great. I'm super excited to have this opportunity to speak with you about what we've been working on in the past about 10 years. My group has been developing an integrated technology pipeline for more holistic, multi-scale molecular imaging and phenotyping of animal and human organs. This technology can be applied to any types of tissues, including liver, but my group has been mainly studying the human brain. And human brain is one of the most complex systems that people are studying. It's probably as complex as liver. It contains 200 billion cells, 100 billion neurons, each making tens of thousands of unique connections with thousands of other neurons. So there are 100 trillion connections in our brain. And this complex system often fails. There are over 600 neurological disorders. And in our lifetimes, we will be touched by any of these neurological disorders one way or another. For example, in our lifetime, 1 in 3 of us will eventually suffer from Alzheimer's disease or other forms of dementia, and the cost to treat Alzheimer's disease patients has been rapidly increasing. It was a couple of years ago over 300 billion in the US alone. And the challenge in studying this neurological disorder is that we need to extract multi-scale information from this large tissue, highly heterogeneous, complex tissue. We need to understand brain-wide connectivity that covers like 10 centimeter length scale local circuits and long-range circuits that's about 1 millimeter to 5 millimeter scale, neural morphology and local connections that's from 10 micron to 100 micron scale, and synapses those are the neural connections, and that's about 1 micron. And these synapses have thousands of unique proteins that determines the functional property of these synapses, and those molecules are on the order of 1 nanometer scale. So to understand this complex system, we need to somehow extract all these multi-scale informations, ideally from the same tissue, but that has been very challenging. And one of the promising technology is fluorescent microscopy, but there are many challenges in applying these fluorescent microscopy techniques to extracting these informations from this whole human organ scale tissue. So to address this challenge, we have been developing largely 2 sets of technologies. One set of technologies engineer tissue physical chemical properties to preserve all these key informations that we care about, structural, anatomical, molecular, cellular informations, while allowing us to use light and chemicals to extract these informations from intact tissue without mechanical section. So to enable that, here are technologies that we have developed, and these technologies engineer tissue optical properties. It makes non-transparent tissue optically transparent. It makes also tissue permeable to molecular probes. We also develop technology that expands tissue fourfold linearly while preserving all the 3D proteome inside the tissue. We also developed a technique called ELAST that makes engineers mechanical properties of tissue, making it stretchable, compressible. And I'll explain why we developed this technology, particularly for human brain tissue. And I will quickly go through these technologies, starting with Clarity. Clarity makes tissue, this non-transparent mouse brain, optically transparent by replacing lipid bilayers with nanoporous transparent hydrogel. So we deliver hydrogel precursor molecules like monomers, initiators, cross-linkers into the tissue everywhere, and then we form dense hydrogen mesh that is porous and transparent. And after that, we tether all the biomolecules to this mesh. And after that, we can remove all the lipids and make the tissue transparent while preserving the tissue morphology, cell distribution, cellular morphology, and molecules inside the tissue. So here's one example. This is mouse brain expressing yellow fluorescent proteins in subset of neurons. We cleared this tissue and we imaged it using fluorescent microscope without any mechanical sectioning. This rendering shows the bottom half of the brain. And as we zoom in, you can see the individual cells, the circular blobs are individual cells, and you can see projections coming out of these cells. So we can clearly map the distribution of these YAP expressing cells and their bulk projection patterns. But this technology wasn't good enough to preserve human clinical samples and also mRNAs. So to address that challenge, we developed a technique called SHIELD. And in SHIELD, we use polyepoxide chemical that looks like my hand, and it can cross-link many molecules, also form intermolecular cross-link to kind of stabilize those molecules. And using this technique, we can preserve all these key informations, including mRNA. And also, it makes some functional proteins hidden chemical resistant. For example, yellow fluorescent proteins and TD tomato, if they are just PFA fixed, they lose the signal after heat treatment. But once they are shield preserved, the signal is well preserved. So using this technology, we can basically extract out many key informations from intact tissue, such as neural projection patterns shown here. So we use virus to express red fluorescent protein and green fluorescent protein in subset of neurons in brain region called GPE. And after that, after mapping the whole brain to see where these neurons are projecting to, the downstream target areas, we next did antibody staining and mRNA fish to extract molecular information from these cells and also downstream target cells. So this is kind of high resolution images of the same brain showing the GPE area, label cells, and their projections. So what we were trying to achieve was mapping these projection patterns of individual neurons that requires tracing, requires high resolution imaging to resolve these fibers and tracing these individual fibers. But at this resolution, the resolving power is just not high enough. So to obtain much higher resolution data set, we use map technique that expand tissue fourfold linearly while preserving all the structural and molecular information. So after map processing, we image the same tissue using the same microscope but now we can see all these individual fibers labeled with red fluorescent protein and also individual synapses labeled with green fluorescent proteins. So using this approach, we were able to map long-range projections of a single neuron shown here. So the neuron in GPE on the left is projecting to many downstream regions. You can see the squiggly lines, they are axonal projections. And this is more kind of map view showing how this neuron is connected with downstream target neurons and also some molecular properties of these neurons. So using basically these technologies that engineers teach physical chemical properties, we can preserve all these key informations and we can extract those informations from intact tissue in a scalable manner. But the next challenge is making this approach more high throughput and scalable. For many applications, we need to deliver molecular probes like antibodies and fish probes deep into the tissue. Traditionally, to achieve that, people have been slicing tissue into very thin sections or dissociate tissue into single cells, so that all chemicals can easily access the cells, and all cells can experience the same chemical processing conditions. But when you have 100 millions of cells densely packed in intact tissue, delivering chemical is a major challenge, and depending on where the cell is located, different cells could experience very different chemical processing conditions. So to address this challenge, we develop technologies that engineers how molecules interact and how molecules move inside the tissue. And I will just highlight two technologies that we developed here. These two videos show mouse brains stained with the same antibodies and anti-carbinding SMI 31-2. And this video shows a series of optical sections. And the right side is a technique called stochastic electrotransport that enhances the transport of molecules by many orders of magnitude using stochastically changing electric field. But that is certainly not enough to achieve a good uniform staining, because the reaction is so fast between antibodies and molecular targets. So we developed another technique called E-flash. In this technique, we use stochastic electrotransport to enhance the transport, and at the same time, we engineer the chemical reaction kinetics. So we start with very slow reaction and gradually increase, so that all cells within the same tissue can reach steady state at any given point. Equilibrium at any given point. So that allows hundreds, millions of cells in intact mouse brain to experience the same reaction conditions. And using this technique, we were able to achieve uniform staining of whole mouse or rat brain-sized tissue just within 1 day. So these techniques are good for animal organs, but they are not good enough for imaging human organs, because human organs are just so much larger than animal organs. For example, human brain is 3,000 times larger than mouse brain. And even if we can completely make human brain transparent, there's no optical microscope that allows us to image this big tissue at high resolution. So we had to develop another technology pipeline summarized here specifically for imaging, multi-scale imaging of human organ-sized tissues. And this technology has three key technology components, the advances. One is megatome that allows us to slice the tissue without losing connectivity information. Amylase technique that allows us to clear this tissue while preserving all the chemicals, and also enable us to deliver chemicals really fast using mechanical pumping, which I will describe in a minute. And also on-slide technology that is computational algorithm that allows us to stitch back these images, 3D images from individual slabs back to whole human brain data set to a point that we can reconstruct projection patterns of individual axons, individual neuronal axons. So I will quickly go through these technologies, starting with megatome. Megatome basically can slice any human organs with amazing precision, and it does not damage the tissue. It doesn't abrade the tissue, and it preserves even axonal connectivity information. So, and the way we achieve this is we vibrate these blades only along this sliding axis, and we do it much faster with much faster frequency and much bigger amplitude. And that allows us to make really clean, nice slices. So here's one example. The top is slices cut using megatome. This is human brain cerebellum. The bottom is cut using Leica vibratome that pretty much most people use. And as you can see, if you look at the heat map on the left, the megatome sliced tissue is very uniform thickness, but Leica vibratome sliced tissue is very uneven. And if you see the morphology of the cerebellum, it is really well preserved in megatome sliced tissue, but Leica sliced tissue, you see a lot of damage. And when we image this cut surfaces after staining, we were able to identify cross sections of cut individual fibers, which is shown here, red and green. Those are axonal fibers, the cut cross section. We were able to match that. And also here is validation using fluorescently labeled neurons. This is before slicing, after slicing. Again, we were able to fully reconstruct the projections after slicing. So now using this technique, we can slice whole human organ without losing information. The next step is engineering these 4 millimeter, 5 millimeter thick slabs into hydrogel. And for human brain slabs, we use ELAST technique. And this technique, again, use hydrogel, make it transparent, but at the same time, it makes tissue compressible and stretchable. So the tissue is virtually indestructible because it's like a rubber. But if you use molecular probes, you can label and image all the molecules and neurons and their morphological patterns and non-neural cells. And this compression and stretching, even you do thousands of times, it does not damage the microscopic structures. So this is before stretching image showing human brain, lectin labeling blood vessels, GFAP labeling activated astrocytes. You can see star-shaped fibers of astrocyte. This is before, this is after, and you can see the morphology is very, very preserved. So why are we doing this? Why are we making tissue compressible and stretchable? Because what we realize is that in order to enhance the transport of molecules into this big human brain slab, if the tissue is compressible, we can actually compress the tissue to decrease the thickness of the tissue. And if we decrease the thickness tissue by 6-fold, you can actually shorten the time required for antibodies or any other probes to penetrate into the tissue by 36-fold, right? So we are basically decreasing the traveling distance required for the probes. And after that, we can just reverse the compression and then image. So using this approach, we demonstrate that we can stain like 5 millimeter thick human brain tissue within just 1 day, which is 100 times faster than conventional method. And we now combined this ELAST with MAP technique to make tissue elastic and at the same time expandable. So the left is the big coronal slab after shield clearing. This is ELAST process. There is slab, it's just too transparent, so you cannot see it. But we can expand that in water for high resolution imaging. So we use this technology pipeline to process 2 tissues. One is from 61-year-old female with no clinical evidence of neurological impairment. The second tissue is from 88-year-old female with Alzheimer's disease. So top is the donor 1 control. The bottom is the tissue from AD donor. And what we first did is we used NewN to label all the neurons to map the 3D density distribution of these neurons in the entire slab. And we identified regions where this AD tissue shows significantly lower density of neurons. To further study this region, we did many rounds of staining using neuronal, subneuronal markers, non-neuronal markers, axonal projection markers, myelination markers, and also pathological markers. The right is AD images. The left is control. The bottom row shows pathological markers. You can see that AD tissue has a lot of beta-amyloid plaque and post-tau. The control tissue also has some beta-amyloid, but very little of post-tau. And you can see the distribution patterns of different cell types and substantial differences in distribution pattern and morphologies of activated astrocyte. So this video shows more details. So top is control that shows red astrocyte patterns, and the bottom is AD brain, and these are showing a post-tau and A-beta distribution, and you can see these details using our approach. And at this resolution, you can correlate, okay, how this pathological vector distribution affects different cell types, their distribution patterns, etc. Also, at this resolution, we can even see the overall projection patterns. You can clearly see that left control and AD, right? The projection patterns of axons are very, very different. Right? In AD, some fibers with different orientations are heavily reduced. The advantage of using fluorescent microscopy is you can change the objective to get high resolution images simply. And at this resolution, we can see morphological details of individual cells like microglia, astrocyte, and neuronal cells and get quantitative information. And this is subcellular resolution imaging. And we can go further. We can expand the tissue and then do very high resolution imaging to image individual synapses. So that's what we did. We labeled pre-synapses. Those green and red dots are basically synaptic protein clusters. The gray is beta amyloid plaque. And this is normal brain. In areas where the beta amyloid plaque, still you see a lot of synapses. So synapse density inside of plaque, outside plaque, there's not much difference in normal brain. But in the AD diseased brain, so I will show those images, the 3D rendering in a second. So this is diseased brain. Now you see that gray labeled beta amyloid plaque has quite different morphology. And you see red and green dots still outside of plaques, but inside the plaque, they are abolished. The synapses are gone inside the plaque. And what was interesting to us is that even in AD tissue, outside the plaque, the synapse density is pretty much similar with that in control tissue. So it shows that the synapses are selectively eliminated within these beta amyloid plaques. So we can do this super resolution imaging to resolve individual synapses. And after that, collecting all this data, what we want to do is reconstruct these images such that to a point that we can trace individual axons. If we can do that in human brain, then we can really map neural projection patterns, connectivity patterns in human brain, which hasn't been possible until now. So we developed a computational algorithm that uses blood vessel distributions to connect stitch back images from different slabs. And after that, we use axonal projection staining to reconstruct the neural fiber connectivity. So this shows the reconstruction of 26 slabs in human brain. So using this technology, we hope to launch NIH Center for Multiscale Multiomic Human and Non-Human Primate Brain Atlases end of this year or early next year. And using this technology pipeline, we will image a larger number of human brains and non-human primate brains to really study, understand individual variabilities in human brain and also differences between human brain and non-human primate brains. And I hope that this technology can be used for studying other organs, including liver. With that, I will finish my talk by acknowledging the group members, collaborators, and funding sources. Thank you.
Thank you, Dr. Chung. We have time for 1 or 2 questions. And we also have some trainee micro talks coming up. So if you're one of those speakers, please come on down to the front. Any questions for Dr. Chung? Well, I look forward. I'm sure we have questions that can continue over later discussion. Thank you so much again for your talk. All right, so our first trainee micro talk comes from Dr. Manjari Trivedi, a postdoctoral fellow in the Goessling Lab here at Harvard Medical School and MGH. And the title of Manjari's talk is Elucidating the Role of Macrophages in Liver Regeneration Post-Injury. Again, a reminder that these speakers will have their posters out in the poster session in just a little bit, so save your questions for that time. Thank you.
Hi, good morning. Thank you for having me here. Liver is a highly regenerative organ. And in when patients with acute Tylenol toxicity get to the hospital, over 90% of their liver is damaged. But a year on, it's completely regenerated. But if the same patient were to be addicted to alcohol, their liver would lose its regenerative capacity and would develop chronic liver disease. And inflammation goes hand in hand with chronic liver disease. So we are studying how inflammatory cells like macrophages can help with liver regeneration and in an acute injury setting, and apply these findings to chronic liver disease as well. In the lab, we use transgenic zebrafish larvae, and we use a chemogenetic system to ablate the liver and cause acute liver toxicity using the drug MTZ. We treat the fish with MTZ for 24 hours and image the livers throughout the injury phase. We then remove MTZ, let the liver regenerate, and image them again. So after 24 hours of MTZ exposure, we see that the liver is completely ablated. And when we removed MTZ, the liver regenerates really quickly in just 3 days. How do macrophages behave here in this injury? So within just 4 hours, there are significantly more macrophages accumulating near the injured liver. And over time, more and more macrophages are coming in. And these macrophages go away only when the liver has regenerated after 3 days. So they must be doing something more important here. So next, we use the same MTZ to ablate the liver and ablate the macrophages, and saw that without macrophages, the livers did not regenerate as well. But they recovered in day 3. Next, we asked, are there different populations of macrophages that may have a bigger role in liver regeneration? So we decided to use clodronate to ablate macrophages either before or after the injury. Here we have a control liver and a liver that had helped to regenerate with from the macrophages. When we ablated these macrophages before the injury, the liver did not regenerate as well. And when we ablated the macrophages afterwards, the livers did just fine. So the first 24 hours after the injury is a key time period for macrophages to do their magic. We also noticed these little holes coming up in poorly regenerating livers. So we followed their progress over a few days and saw that these holes became worse and bigger. So without macrophages, the livers do not regenerate well and start accumulating structural defects. So today I've shown you acute liver toxicity, the liver is damaged, different macrophage populations respond to it, and the liver regenerates well. But if we get rid of the early response macrophages, the liver does not regenerate and rather accumulate structural defects. Thank you.
Thank you very much, Manjari. Our next speaker is actually rescheduled to the 3rd session, so stay tuned to hear from Dr. Nagesh later today. Our next speaker is Dr. Urmila Jagtap, a postdoctoral research fellow in the laboratory of Frank Slack at Beth Israel and Harvard Medical School. Thank you.
Hi. So, do you ever feel guilty about eating that slice of pizza that's of your favorite, and maybe the junk food that keeps you excited about? I certainly feel very guilty, and the reason is this. According to the American Liver Foundation, 25% of adults in the United States suffer from non-alcoholic fatty liver disease. Now, if this statistics is true, every 4th person in this room is already having fatty liver disease or is going to get one very soon. Now, that's scary. I know, because I am one of those. So what goes wrong? If you see the hepatostat, which is a homeostatic condition in liver, which is very critically maintained. And when this balance gets perturbed, the healthy liver gets pushed to the disease condition, starting from fatty liver to NASH to cirrhosis, and in extreme conditions, hepatocellular carcinoma. As you can see, there are multiple factors that maintain this critical hepatostat, and one of these factors is microRNAs. Now, microRNAs are small non-coding RNAs, which though are very small in size, they have major metabolic functions. One of such microRNAs is microRNA 21, which is my favorite. And the reason is because it has been shown to have major effects in all the liver-related pathologies as well as metabolism, like fibrosis, fatty acid uptake, glucose metabolism, etc. However, all the attempts to use microRNA 21 as a diagnostic or therapeutic intervention have not been very successful. So what am I doing about this? In my research, I'm trying to create a comprehensive model of liver disease and hepatocellular carcinoma using high-fat diet as an injury in mice. Using this model, I'm trying to understand the role of microRNA 21 in high-fat diet-induced liver pathogenesis. And I'm also trying to investigate microRNA as a potential therapeutic for this progressive liver disease. To do that, I'm using wild type and whole body knockouts of MIR21. To kickstart the liver injury, I give them a small dose of carcinogen. And for the chronic liver insult, I either feed them with chow or high-fat diet. Over the course of 32 weeks, the wild-type mice gradually shows symptoms of metabolic dysfunctions as well as liver pathogenesis like NASH fibrosis and hepatocellular carcinoma. Interestingly, just the knockout of MIR21 was able to show in our study all these pathogenesis phenotypes, though slowly but surely. And most importantly, what we observed was that the lack of MIR21 and the combination of high-fat diet was able to not just accelerate this progression much faster, however, it was also much more severe, indicating that MIR21 is having a protective functions towards progressive liver disease. Now, using a lot of in vivo and in vitro experiments, we tried to next understand how MIR21 might be exerting its effects on liver metabolism. And we came down to this particular target gene, which MIR21 directly interacts, which is TGF-BI, that is TGF-beta-induced gene. And that is how MIR21 is exerting its protective functions by negatively inhibiting it. Now, the patients go to the doctor only when they have developed the disease, right? So to use microRNA as a therapeutic, we let the wild-type mice develop the disease over 16 weeks of time. And then at that point, we injected the mice with either negative control or MIR21 mimics subcutaneously. And what we observed that the mice that were injected with MIR21 mimic are not just able to take care of the metabolic dysfunction phenotypes, but also later late stages of liver pathogenesis like fibrosis and hepatocellular carcinoma. So together, what we have done in this study is that we have created a comprehensive model of liver disease and HCC, which shows pathological phenotypes that are observed in human patients. We have seen the protective function of MIR21 towards this pathogenesis, and we show that these effects are majorly regulated by the interacting target of MIR21, that is TGF-BI. And most importantly, we've been able to show in our work that MIR21 could have a potential therapeutic value to treat the progressive liver disease. Given that the Nobel Prize of 2024 was awarded to the discovery of microRNAs and another amazing demonstration of RNA therapeutics in COVID-19 pandemic, we believe that the use of microRNA therapeutics for the progressive liver disease is not very far, and we are very hopeful that we are trying to go in that direction just by contributing a little bit from our lab here. Thank you.
Thank you. And our final speaker today is Dr. Emeli Chatterjee, a postdoctoral researcher from the lab of Saumya Das at the Cardiovascular Research Center at MGH. Welcome.
Hello, everyone. So as we all know nowadays, fatty liver disease or MSLD is currently a growing problem worldwide. And not only that, closely 30% of the adult population is currently affected by this particular disease condition. And in addition to this, we can also say that this MSLD condition is directly associated with the development of several cardiovascular diseases, which may include the heart failure. So there should be some kind of inter-organ crosstalk is going on between heart and liver. So being an extracellular vesicular biology lab, we were also interested to see whether extracellular vesicles or EVs are playing any role for the development of the cardiovascular disease condition during the MSLD. Now, as we know that extracellular vesicles or EVs, which serves as a tremendous player for the inter-organ crosstalk, as it can carry different types of cargoes like lipids, proteins, and nucleic acids from the donor organs to the recipient organs through the circulation. So in that respect, we were also interested to see whether liver-derived EVs are playing any role for the development of this cardiovascular disease. So in our current case, what we have done, as you can see here, that we have utilized a quad liver chip culture system. And utilizing this particular platform, we have developed our MSLD model. And eventually, we have isolated the EVs from these MSLD disease condition, and then we have treated the iPSC-derived cardiomyocytes with these MSLD liver-derived EVs. And then we were interested to see what are the effects of this EV treatment on the cardiomyocytes. So first of all, we have characterized the cardiomyocytes, and then, as you can see here, we have checked the transmission electron microscopy of those cardiomyocytes. And with the MSLD EV-treated group, we can see there is a huge accumulation of the lipid droplets within the cardiomyocytes. And not only that, from this metabolic gene expression, we can see that the overall fatty acid transport was increased, but the overall utilization level was not properly done. So to get a more insight for this, we have done transcriptomic profile and as well as the metabolomic profile. So from this transcriptomic profile, as you can see here, that we were able to identify several differentially expressed genes. And not only that, from the pathway analysis, we are able to detect several disease-relevant pathways. And from this metabolomic profile, you can see that how nicely the control EV-treated group and the fatty acid-derived EV-treated group are nicely separated from each other. So our next question that came in our mind, that what are the difference between these control liver-derived EVs and these MSLD liver-derived EVs? So we have characterized the EVs. So for the initial part of the characterization, we have done the biophysical characterization via some high throughput microscopy. And interestingly, we have seen from this microscopy that exclusively the hepatocyte-derived EVs number and the size was significantly higher in the fatty acid-treated group compared to the control group. So we mostly focus on the hepatocyte-derived EVs, and then we checked the transcriptomic profile of those particular EVs. And here, as you can see, that from this smaller end transcriptome, we were able to detect several differentially expressed microRNAs, and eventually we were also able to detect several impacted pathways. As you can see here, that some of these top 5 pathways were actually disease relevant. So, as a nutshell, we can say that there are some definite existence of the liver-derived EV-mediated inter-organ crosstalk between liver and heart during MSLD condition. And it is also possible that targeting this EV cargo might mitigate the overall heart failure impact. I would like to thank the organizer and my lab members and my PI, Dr. Saumya Das. Thank you all.
Well, thank you so very much for the invitation to speak. I am so delighted to join you, albeit from clinic. It's my clinic day, which was what precluded my ability to be there in person, but an absolute pleasure to be here. And I hope to spend the next 20 minutes talking to you about pediatric liver tumors and convincing you that they are the perfect paradigm for collaboration in a fascinating series of diseases. I have no disclosures. So my objectives are to give you a brief overview of the most common tumors affecting pediatric patients in the liver, talk a little bit about ongoing collaborations, and then end with a discussion of novel therapies, where we are currently and why we need more for this population of difficult to treat patients. So let's start with a brief overview of disease. Why am I so interested in liver tumors, and why should you all be in pediatric patients? When we think about the spectrum of diseases that affect pediatric patients, liver tumors exist on the far right. They comprise only about 1% to 2% of all tumors that affect children, but they are the perfect paradigm for collaboration in a multidisciplinary sense, which I hope to convince you of. When we think about the most common tumors affecting pediatric patients, we think first by age, and then categorize tumors in both the benign and the malignant states. For the purposes of this talk, we'll primarily focus on hepatoblastoma, HCC or hepatocellular carcinoma, which is a much different disease in children compared to that in adults, and fibrolamellar carcinoma, which previously was lumped in with conventional HCC, but is an entity all its own. Briefly, hepatoblastoma is increasing in incidence. There are about 2 cases per million children per year now. Hepatoblastoma accounts for about 2/3 of all liver tumors, is the most common liver tumor that affects children, and accounts for about 90% of tumors in children under the age of 4. There are few associations with familial conditions like familial adenomatous polyposis or overgrowth syndromes, but the greatest risk factor is low birth weight associated with prematurity, which may be why the incidence globally is increasing because more and more children who were born prematurely are living to an older age. And we know that the risk increases nearly 20-fold in children born at less than 1,000 grams at birth. The SEER monograph can show you that incidence drops off appreciably after about 4 years of age. So this is a diagnosis of very young children. Conversely, conventional HCC in children has about an incidence about half that of hepatoblastoma, and it historically varied depending upon Hep B vaccination rates. So in sub-Saharan Africa, Southeast Asia, where Hep B vaccination rates lagged, incidence of HCC was higher due to pediatric cirrhosis of the liver. But now that vaccination rates have, in essence, leveled out globally, the incidence rates are pretty level across all continents. This is a disease predominantly of adolescents and young adulthood, not so much in the younger age population, although occasionally we do see it in young children. Hepatoblastoma is treated with a backbone of cisplatin and doxorubicin. Those are the mainstays of therapy. We achieve excellent outcomes for patients with what we perceive to be low or standard risk disease. That is historically a disease that's easy to resect surgically. For patients with high-risk disease, often characterized by metastases, these children are very difficult to cure. When we think about conventional HCC in pediatric patients, histologically under the microscope, it may look similar to the disease seen in adults, but only about 20% of the cases in children arise in the context of cirrhosis. That's vastly different from the disease that arises in adults, wherein 80% of adults have cirrhosis, either due to alcoholism or viral hepatitis, etc. Because we think the disease is different in children, we know that those children respond better to chemotherapy than adult HCC, and we believe it to be on a different genomic spectrum, which I'll introduce you to a bit later. Fibrolamellar carcinoma, the third of the most common tumors affecting pediatric livers, is very unique from conventional HCC. In the last decade alone, we have learned that these tumors are characterized by a DNA JB1 protein kinase A fusion protein that then leads to aberrant function of protein kinase A. These tumors also arise in a completely normal liver, and there's no agreed upon systemic approach to therapy, and we know that surgery is crucial, but unfortunately, patients typically present in the advanced stage and are very difficult to cure. What we know, independent of histology, is that there are unifying requisites for cure of these diseases. We need adequate systemic disease control and response, and we need to resect all sites of disease. So then how are we doing? So for patients with more advanced disease and hepatoblastoma, this study that recently came out of Europe showed that we can cure about 80% of those patients. But this study included patients that had disease throughout the liver and those with metastatic disease. So the non-metastatic patients kind of pulled up the curve, when in reality, patients who fail to clear their lung metastases only survive about 50% of the time. Pediatric HCC has a dismal overall survival. Only about 20% of these patients live, and those are the 20% of patients who incidentally had disease diagnosed before it spread. So clearly we need to make more progress. So what about the biology of these diseases? Well, we're learning they're on a spectrum. So we know that they're genomically silent tumors, particularly hepatoblastoma. So if you think about the spectrum of somatic mutation rates across cancers, many of you have probably seen figures like this. Hepatoblastoma lives on the far left. Adult HCC lives somewhere in the middle, and pediatric HCC someplace closer to the left end of the spectrum. We know that the Wnt-beta-catenin pathway is crucial for these tumors. It's like a sine qua non of disease. 10% of patients don't have aberrations in Wnt-beta-catenin, but the vast majority do. And we know that these aberrations in beta-catenin destabilize this complex, leading to free beta-catenin, which goes to the nucleus and affects transcription, translation, and cell proliferation. The abnormalities in beta-catenin are very wide in spectrum. There can be point mutations, small deletions, large deletions, but we know that this is a driver of disease. But we also think that these diseases that I just relayed to you, at least hepatoblastoma and hepatocellular carcinoma, exist on a spectrum in pediatric patients. And when they occur, depending on patient age, development of the liver, likely development of the immune system, and/or additional insults, probably dictate what type of disease the patient develops. This figure, which I took from a paper by Pavel Sumasin, published in 2017, it's a pediatric group at Texas Children's, postulated that perhaps there's a unique stem cell that either goes on to look more like an hepatoblast or progenitor cells, has a few kind of benign mutations and looks primarily like fetal liver, versus the stem cell may go a different route, gather additional mutations and look more like a fetal epithelial hepatoblastoma, which is the vast majority of diagnoses. That being said, there's a new diagnosis recently described called hepatocellular neoplasm not otherwise specified. Under the microscope, this looks like a hybrid between hepatoblastoma and HCC, usually occurs in older children and has biologic features that are more aggressive on the end of the spectrum, which can include TP53 aberrations, TERT promoter mutations, etc. There have been published genomic signatures on these tumors, first from our European colleagues, and then kind of recapitulated by our colleagues at Texas Children's. But essentially what these groups have showed that when they looked at genomic profiles and expression panels, that there are two subsets of patients, the C2 subset that has a poor prognosis and the C1 that has a more favorable prognosis. The genes that are expressed in the C2 category are characterized by an increased risk for proliferation, invasion, etc. And when our colleagues at Texas Children's recapitulated this work, they found something similar. When they did kind of hierarchical clustering of expression profiles, they found an HB2 category, much like the C2 aggressive group of patients, an HB1 category, much like the C1 category. But the realities are, we know that these patients cluster clinically. We just don't know what to do about the high-risk patients in terms of getting them over the finish line to an adequate cure. Recently, our Japanese colleagues published a really nice paper in Nature Communications that essentially described, again, with both DNA and RNA sequencing arrays, 3 different cell types: a proliferative cell type, much like the C2 subtype, a mesenchymal subtype that was intermediate in aggressive nature, and then a very benign subtype, again, much like the C1 grouping. But they also went on to look at the epigenetic patterns of these tumors, and they were the first to do so on a relatively larger scale. They demonstrated preferential methylation of paternal alleles during carcinogenesis. They looked at kind of a methylation of the IGF-2 promoters and showed that aberrations here were more linked to immature cells in the fetal liver. And then when they looked further at methylation of promoters and enhancers, they again subcategorized patients into an HCN-NOS or more aggressive category, a middle-of-the-road category, and then a more benign-like category. And taken together, they showed that we think is happening is that, again, depending on when these patients are diagnosed, the state of their liver and the number of genomic abnormalities and/or epigenetic abnormalities they accrue dictates the type of tumor that develops. Pediatric HCC, I mentioned, we believe to be very different from adult HCC, and there's only one study to look at this, which essentially showed that deletions in CTNNB1 are more common in either hepatoblastoma or conventional HCC than they are in adults. But that adult tumors tend to have a much higher rate of promotion point mutations in the TERT promoter region or loss of function mutations in TP53. So we know they're different. We know the context in which they arise are different, but then how do we bridge the gap in treating these patients? And finally, we studied a small series of patients with pediatric conventional HCC with immune checkpoint inhibitors. Checkpoint inhibition is kind of the mainstay of treatment for adult HCC, but is not well studied in pediatric HCC because, again, we don't really believe them to be similar tumors in nature. However, I will say of this small cohort, 1 patient achieved a complete response and 2 prolonged stable disease. And when we looked at bulk RNA sequencing, we found that the responders to immunotherapy had decreased expression of genes associated with T cell dysfunction, as you might suspect, immunosuppression, inflammation, and cancer growth and invasion. So there's different types of pediatric HCC, although these patients were all of a similar age, so still difficult to tease out who might respond and who might overlap more with the adult phenotype. I wanted to kind of segue into what collaborations we have existing to learn more about these tumors, and then how we think about novel therapies and where to go next. So one of the biggest advancements we've made recently is study of these tumors on an international global level. We launched what's called the FIT trial, which was conducted in the Children's Oncology Group in the United States, all across Europe and all throughout Japan. And the goal was to harmonize the way we studied these tumors, both hepatoblastoma and for the first time prospectively, PDHCC, to genomically validate some of the signatures I already showed you, and then to create a really large biologic repository of tissue and serum samples from all of these tumors. We accrued over 500 patients in the United States, which for us in pediatric oncology is a huge number. These are rare diseases that are difficult to treat in a holistic and kind of uniform way. And then globally, we recruited over 1,100 patients. So this is going to serve as the largest source of data we'll ever have access to for patients with these rare diseases. Similarly, we wanted to learn more about relapse because when these patients relapse, they're very difficult to salvage and cure. So we launched what's called the RELIVE Registry for relapse. This is a global initiative intended to collect data on how individuals are treating these patients, either with relapsed hepatoblastoma or HCC. And ultimately, this platform will allow collection of relapsed tissue and serum samples as well. Finally, in the United States, we launched something called the Liver Tumor Research Consortium. This is a collaboration between us in Dana-Farber and colleagues in Cincinnati. We kind of operationalized it, collecting retrospective data for the RELIEVE initiative that I just talked to you about. But now prospectively, we're launching two prospective registries that will allow us to learn more about relapsed hepatoblastoma and PDHCC in real time, one of which I'll spend a little more time talking about. And finally, at Dana-Farber, we launched in the last 5 years the Liver Tumor Center of Excellence. This was intended to provide patients and families a single point of patient entry, concierge care for these rare diseases with multiple experts within the multidisciplinary care community, and a pipeline for clinical care, trial offerings, and research samples. We've more than tripled the number of patients we see annually, and when you think about the incidence rate of these tumors globally, we see a large fraction in the United States and in the Northeast alone. So what about translational collaborations? The first one I'll talk about is the patient-centric initiative that we're working on with the Broad Institute called Count Me In. Some of you may be familiar with this, but this is a patient-initiated consent for submission of medical records and serum, tissue, and urine for a deep dive genomic sequencing opportunity, with the data generated being ultimately shared publicly. We're thrilled that they were invested in a pediatric HCC CMI initiative. And our goal is to not only consent families throughout the United States who are actively undergoing treatment, but to likewise access the HCC samples from the COG trial and perform similar genomic analyses on those so that we'll have the largest cohort to date with more information about how these tumors behave, how these patients were treated, and what types of novel therapies we can think about next. We've launched an imaging initiative with Gary Schuler, who's a radiologist at Cincinnati, whereby we're looking at radiomics for the first time in pediatric liver tumors. This is a pixel-level analysis of tumors using radiographic imaging, either MRI or CT. Right now, we're in the early phases looking at tumor segmentation, comparing human approaches to AI-generated approaches. This process was initiated first with hepatoblastoma, ultimately will be translated to hepatocellular carcinoma, and the goal is that hopefully this technique will aid with diagnosis and an eventual prediction of response. We've launched a collaboration with Brian Crompton, who many of you may know through the Broad Institute and Dana-Farber. He's looked at circulating tumor DNA across an array of pediatric diagnoses and has recently engaged him to study pediatric liver tumors. We know these tumors are very heterogeneous. They're embryonal tumors that have a mixture of different tissues indwelling. And biopsy alone doesn't really do justice to the fact that these tumors are heterogeneous, and we may not capture the full genomic spectrum of abnormality with a single needle biopsy. Our hope is that ctDNA will give us more insight to tumor heterogeneity. We'll be able to access molecular and methylation profiling and pair that with annotated clinical data. And our thought is that there will be both therapeutic and prognostic implications, as well as a detection of early relapse patterns utilizing this approach. When I last spoke at MIT, which was many, many years ago, about our work in liver tumors, we aligned with Salil Garg, who's now at Yale, and we started a single-cell sequencing initiative wherein, again, we hope to better assess tumor heterogeneity for these samples, better understand cell of origin across the spectrum of diseases, and then get some insight as to the immune infiltrate. So we have a large cohort of patient samples that we were able to collect viably frozen, so that we can assess anywhere from the hepatoblastoma, HCC-NOS, and HCC end of the spectrums at diagnosis and throughout the course of therapy. And we're excited to see what kind of data we generate through this initiative. And finally, my pie in the sky goal is to create what we aim to call the Liver Tumor Accelerated Platform for Pediatrics. This would be the ideal opportunity to better study model design and novel drug testing. We know that these tumors are very, very hard to grow. They're hard to grow in vitro. They're even harder to grow in vivo. But there are groups throughout the United States that are engaged in either generation of cell lines, organoids. There's a quail egg model, mice models, and then, of course, Wolfram and others working on zebrafish models. And our goal is really to align with industry to cycle novel compounds through more than one model. We know that one model never really translates efficiently to patient success. And so our goal here was to study these rare diseases that are difficult to grow in more than one context. And in the last minute or two, I'll touch on novel therapies because we are making a lot of progress, but I think there's so much more to learn. So there are a number of clinical trials currently open and enrolling for patients with relapsed or refractory hepatoblastoma. Among these are trials studying HDAC inhibition in concert with standard of care chemotherapeutics, with the concept that better access to the DNA prior to treatment with cisplatin or other derivatives might make these chemotherapeutics more effective. There's an ongoing trial studying a drug called tegavivint. This drug is intended to stabilize the beta-catenin complex, thereby rendering what we believe to be a driver of these tumors ineffective. There's an open trial studying an MDM2 MDMX inhibitor because there's preclinical data in hepatoblastoma suggestive of the fact that TP53 may play more of a role than we realize. And finally, there's a lot of interest in GPC3. This is a protein expressed on the cell surface of liver tumors explicitly, not normal liver. And this antibody, codrituzumab, is an anti-GPC3 antibody intended to bind specifically to tumors and hopefully engage immune cells. In the realm of HCN-NOS or HCC, there are also a lot of immunotherapy trials that are currently ongoing. And as we know, pediatric patients have been less apt to respond to immunotherapies than adult patients, but there are exceptions to that rule. So we are studying checkpoint inhibition in HCN-NOS and HCC. I recently aligned with Eureka Therapeutics, a small industry sponsor, to write a trial for a TCR mimic that binds AFP peptide. AFP is a tumor marker expressed by nearly all of these tumors in the serum. And a peptide of which is bound to HLA2 can be targeted by this TCR in concert with GPC3. There is a trial out of Texas Children's studying a CAR to GPC3 that's armored. And finally, there's a lot of novel work for fibrolamellar carcinoma. That's the unique subset of disease characterized by that translocation, and that is targetable. So the team in Hopkins has studied and is studying a peptide-based vaccine targeting that chimeric fusion with dual checkpoint, and a unique trial that they just launched that tries to interfere with the metabolic pathways intrinsic to this disease in concert with checkpoint inhibition. So what's our next collaboration? I am convinced that there's more to study. I'm excited that we have access globally, in truth, to one of the largest repositories of pediatric samples to date. And I have no doubt that there's a lot to brainstorm. So I would love to become even further engaged in the research community related to pediatric tumors, because I think there's a lot to learn and a lot that can still be done here. So please, if there are interests, please email me. And with that, I'd just like to thank our Liver Tumor Center of Excellence here at Dana-Farber Boston Children's, the Children's Oncology Group, and all of our global collaborators, and then most notably patients and families, without which we couldn't do half the work we're invested in doing. So thank you again for this wonderful opportunity to speak, and I'm happy to take any questions. I also can't hear anyone if somebody's saying anything.
Ellie, it's Wolfram. That was a really cool talk. Can you enlighten us a bit more about the PDHCCs and the fact that they're non-cirrhotic? And I know you showed some of the molecular alterations. You know, I know that obviously fatty liver disease in the pediatric population is a rising risk factor, but do you know, or do we know anything about other, you know, epidemiological risk factors for these children who develop this form of HCC?
I wish we did. So in truth, I mentioned that 20% of pediatric patients have cirrhosis, but those are patients born with metabolic abnormalities, things like Alagille syndrome, so structural abnormalities. Very rarely do we see a viral related cirrhosis, but a lot of the times those are better differentiated tumors. The well-differentiated tumors occur more frequently in the patients that have a genetically inherited syndrome that leads to liver dysfunction. So of the 80% that occur de novo, their livers for all the world look completely normal. We always biopsy adjacent normal, and we tend to genomically profile both the tumor and adjacent normal when possible. And adjacent normal doesn't tend to have fatty infiltration, at least not in the cases we've seen. So while a potential risk factor doesn't seem to play as large a role as maybe we think, besides the fact these patients are developing disease at such a young age, they haven't really lived long enough for that insult to take effect. We know that, in the cirrhotic environment, there's a lot of inflammation, fibrosis, kind of immunosuppressive cells. We don't see any of that in pediatric tumors. And so of the pediatric HCCs we see some recurrent alterations in KMD2A, ARID1A, sometimes TERT promoter, sometimes the MEK-ERK pathway, but they're one-offs. There's nothing that unifies the diagnoses across the spectrum. So I think we still have a lot to learn. We also know that a good proportion, at least 2/3, have nuclear beta-catenin abnormalities, similar to our hepatoblastoma patients and different from a lot of the adult patients. So again, back to them being on a spectrum of sorts, which may relate more to the age at which insult occurs, as opposed to a unifying genomic factor apart from CTNAB1.
I don't think we have any further questions at this time. For the sake of time, we're going to cut it off here. But thank you so much, Dr. O'Neill.
Thank you so much. It's a pleasure.
Cool. I will now introduce Dr. Li Jianhui, who is the PI in the Shanghai Institute of Biochemistry and Cell Biology. He's currently Assistant Director of this institute, board member of Chinese Association of Cell Biology, President of Chinese Association of Cell and Gene Therapy, editorial board member of Cell Stem Cell, Hepatology, and more. Dr. Hui studies molecular pathology of liver diseases with a focus on cell identity conversion, that is, transdifferentiation and dedifferentiation, in liver regeneration and tumorigenesis. His team demonstrated the conversion from fibroblasts to hepatocytes in vitro, thereby providing one of the first pieces of evidence of transdifferentiation in mammalian cells. His team is also among the first to report cell identity conversion in liver injury and repair. His team, based on these fundamental findings, his team generates functional hepatocytes, high HEP, and pro-LHH for clinical applications. Innovative therapies based on these cells, namely bioartificial liver devices and encapsulated liver organoids, are now being tested in clinics. Dr. Hui is also the founder of Hexael Biotech, which aims in helping liver disease patients with cutting-edge cell therapy technologies. Dr. Hui has received several awards, including 10 top breakthroughs in science and technology of China, China Young Scientist Award, and Natural Science Award of Shanghai Municipality. The floor is yours.
So, how can I start? Yeah, thank you for the organizers, Sangeeta and Wolfram, for the chance for the invitation, and really happy to have the chance to talk here. So, how can I start my slides? I click somewhere.
One of these two.
Oh yes. Oh, okay.
Okay, that works.
Okay, great. Yeah. So I'll talk about cell identification and liver regeneration and cell therapy. So I think for this audience, I don't need to introduce too much about the liver. And the liver is probably the most regenerative organ in human body. And but after repeated injury, I think the liver still will lose their regenerative capability. And eventually, many of this disease will lead to fibrosis or other kind of end-stage disease. And for most of these end-stage liver disease, I think there's no effective therapies. And I think in the field, back to many years ago, people already think about cell-based therapy. So I just named two techniques which we are working at also in the lab. One is so-called artificial liver device. It's an extracorporeal liver supporting system. So in this case, you take the blood outside of patient in end stage of liver disease, and let the blood go through this bioreact, which filled with functional hepatocytes there. And after this, you detoxificate the toxic stuff in the blood and also secret nutrients, other growth factors back to the patient in a way to support the patient, either to bridge it to the liver transplantation or kind of support the function, and the patient, the liver will kind of recover. And there's another strategy, is so-called also topical liver hepatocyte transplantation. In this way, you infuse the cell to the liver, and these cells will engrafted into the liver and then repopulate the liver. So these are the two strategies that have been studied for many, many years, I will say. But for those studies, a very critical issue is you need this functional and expandable human hepatocytes. So when we're talking about the human hepatocytes, it's a kind of paradox, I would say, because in one way, the liver, human liver or other animal livers, they have a very strong regenerative capability. But if you take the cells out and you put them into a petri dish, they don't proliferate. For a long time, people don't know how to handle with these cells. So you cannot always get cells from the donor organs. So this is really a technical issue and a very important biological question, I would say, to address. And this is a beautiful review from Sangeeta a long time ago. She summarized the progress in the field. And I don't go to all the details. There are a lot of efforts in the field. People try to generate functional and expandable hepatocytes. And I will show, actually, our lab also joined this kind of effort a long time ago. So the first thing we did was like more than 15 years ago, we converted the fibroblast cells into we call high-hep cells. So these are human umbilical-derived fibroblasts, or you can get fibroblasts from other tissues. And by overexpression, FOXA3, HF1 alpha, and HF4 alpha, 3 transferrin factors enriched in hepatocytes, then you can convert these fibroblasts into hepatocytes. You see the dramatic change of morphology. They are more epithelial-like cells. And we also published a long time ago, they have also multiple functions of primary human hepatocytes. So then we use these cells for bioartificial liver device. I just quickly go through what we have done. Basically, first we prove concept to use mouse tail tip fibroblast, convert into high-hep cell. Then we did this high-hep cells 3 years after. So take these high-hep cells. I'm working in Case Chinese Academy of Science. Case very focused on translational research, and they give us a big grant to convert to them into a ball, translate them into ball technique. So they will perform the first large animal experiment using high-hep cells as a seed cells for ball. And then we, after this, we want to perform clinical trials. Then we realize we need to transfer the technique to company, otherwise we cannot do it in the lab. So we transfer to the HEC cell, where I'm one of the founders. Then in 2019, performed the IIT experiment, was IND enabled 2 years ago. And phase 1 was finished this year, early this year. They are now entering the phase 2. I think this is goes through like almost 15 years to do all these things. And I think like I'm still waiting the phase 2 data, which is a controlled experiment. But I'll say like for the ball system, you don't really like transplant these cells into the patient. It's a extracorporeal device. So I'm always fascinated, like maybe we can use the cells for transplantation. So in the lab, then at that time, I think like because you see the liver regeneration all the time after you do partiectomy or injury, other chemical injury, all these things. So I think there must be something where you're still missing in to understand the liver regeneration after injury in vivo. So then we at the 2000, actually we reviewed the whole field, like just to put what we have seen a long time ago. So there are a lot of ways to regenerate the liver hepatocytes. So each arrowhead, you can see there's at least one literature published in one paper published in the literature. But I will not go through all these details. I will highlight today the topic of my talk, dedifferentiation. So dedifferentiation means when I talk about differentiation in my talk, it means mature hepatocyte converted into liver progenitor-like cells. So I'm saying like they are not progenitor, exactly progenitor in fetal development. They are progenitor-like cells. So this is go back to the long time ago, was year 2013. Ben Stenger at that time, he published a paper. In that paper, he injured the liver with DDC, a chemical which causes the cholestasis injury. And in this injury, he found a mature hepatocyte, which is all HF-alpha positive. HF-alpha is the transcript factor, mass transcript factor in the mature hepatocytes. So you see they are all positive for HF-alpha. Then he found at the same time the cells, after injury, you see some of the cells express also SOX9. SOX9 is a marker for cholangiocyte and progenitor cells. So in that time, he's very interesting. He publishes a paper, and we immediately noticed this paper because at that time we were looking for in vivo evidence of dedifferentiation or transdifferentiation. And Ben Stenger at that time, he thought this more probably is transdifferentiation because he said like their cells are not expressing AFP, alpha fetal protein. And we are very interested about this, and we have a different idea. We think these cells probably are progenitor-like cells. This dedifferentiation instead of transdifferentiation. So then we look at these cells more carefully. We isolate the cells. I'll say not only our groups, there are also other groups make a lot of analysis of these cells. So what we found is these cells, these HF or SOX9 double cells, they express not only SOX9, they express a whole bunch of genes related to fetal hepatoblast. We name these genes as RG gene. So it apparently the cells are not simply express SOX9. They are acquired already the expression features of progenitor cells. And then we make a lineage tracing of these cells using this very complicated combination of different tools. But the labeling is very specific. So usually you'll see around 5% of these double positive cells after you injure the DDC. Then we will ask what happens to these labeled cells. What we see is they contribute to almost 20% of newly formed hepatocyte, which means they have really stronger regenerative capability compared to normal hepatocytes. Another very interesting finding is these cells not only gives rise to hepatocytes, as I show you these red hepatocytes, they also generate cholangiocytes, as you see here. There's a cholangiocyte here. So which means they have at least, for some extent, the biopotential during after the injury to repair the tissues. So then we also look at whether we can find evidence of these type of cells in the human disease. So we take the primary biliary cholangitis and also HBV infected patient samples and the NAFLD, now people call it MASH samples, and you see all these double positive cells. And recently, there's a Nature paper by Ludwig Valerie Lab group. They demonstrate in the MASH livers, you can also identify these double positive cells, and in the organoid, they demonstrate that these cells has progenitor-like features. So all these data collectively demonstrate in either mouse or human, after injury, there's a type of cells or a type of cell state, I would say, it's because it's not always there. So it's these cells, so HF-alpha, SOX9, double positive cells, they have more strong regenerative capability. They can differentiate into bile duct and hepatocytes. So then we have the idea, so can we expand these human hepatocytes by dedifferentiating them into these progenitor-like cells, this double state positive or progenitor-like state, because these states really gives a very strong proliferation capability. So to that end, then we screened a lot of medians in the lab. I would say like almost 20 different medians, and we only focus on human hepatocytes. So like human hepatocytes, they don't proliferate, as I show you by this BRDU incorporation experiment. The harder you detect BRDU positive cells in traditional culture median. But when we found this median, Wnt3A dependent median, of course, it's not only Wnt3, there are also other components, but the most critical component is Wnt3. Without Wnt3, there's no proliferation at all. This is very clear. So without other components, which I didn't show you here, because all these are published, so there are some kind of proliferation. So you see dramatic difference. 20% of cells are in proliferation status. So you can, if you quantify the proliferation of these cells, they proliferate at least at that time was 7, 8, 9 passages, and you can expand 10,000-fold. And if you look at the control, they are like usually like a P1 or hard to go to P2. It's flat. Okay. So nowadays in the lab, we further optimize the condition. We can now expand the cells for 1 millionfold. So it's almost like I say like if I get one liver, I probably can use it for my treat the whole patient in my whole life, I guess. So then we because we thought like these cells are like they differentiate. So when they look at the expression profile of these protein-HH, so here is protein-HH. So this is the primary human hepatocytes in this PCA analysis. They sit here, and this is the progenitor cells from the embryo liver. So they sit here. And during the culture of these protein-HH, you see they slowly move close to the progenitor cell. They focus on PC1 axis. So in the culture, they never reach to progenitor cell in the fetal, but they are like in the middle. So we call it like biphenotypic cells. And because they are progenitor cells, so they lose a lot of liver functions. So then we asked, can we mature them by some kind of techniques? And in long time, we have already now in the lab, when we culture primary hepatocytes in a 3D condition, they are more mature compared to the 2D culture. So we say maybe we can put these high protein-HH in a 3D culture condition. So we take the low attachment petri dish and force them to form these aggregates. Nowadays, people call them organoids. So you see like here that you see like this, they form a canalicular, very beautiful canalicular. This is tight junction here, cell. And they also have this microvilli on the scan electron microscopy. And this is also, they have enriched over this mitochondria here, very clear. And if we look at the expression of mature function genes, you see here, so in this protocol, you put them into 3D organoids and you culture them into a median called a mature median. And here you see this is primary hepatocytes, and this is 2D cultured protein-HH, and this is organoids, protein-HH from organoids. You see clearly they get all these function genes re-expressed again. Not as good as the primary hepatocyte, but this is really in our hands. We see the most mature hepatocytes, and also we see like these SOX9 and the CD133 expression are reduced after we form 3D organoids. So then the question is, can we use these cells for transplantation? So if you think a mouse experiment, it's also apparent you can do that. So if you still remember the first slides of my talk, this green and red liver, it's a protein-HH transplanted in red. We stain them in red into a FH mouse. So in mouse models, very beautiful. You transplant this repopulation great. But this you cannot translate into clinics. We tried so hard. I talked to almost everyone in the world, like who work on cell transplantation, So there's no precondition protocol so far, efficient precondition protocol to hepatocyte transplantation. Because if you think that at that time, you don't have the expansion per technique, but still you can get a lot of human hepatocytes. You still can do the transplantation in patients. If that works, you will see it already, right? So I think that the field still waits the technical breakthrough to really transplant hepatocyte to the liver. But so seems like we are developing a technique that we cannot use for the translation. But actually, there are also other techniques that people are developing. So there are a long time ago, people think like maybe you can use you take the primary hepatocytes, then you encapsulate them with alginate gel, hydrogel, so that you prevent the immune cell attack to these cells. And you transplant these encapsulated human hepatocytes intraperitoneally, and through in the intraperitoneal, through acid cells, they have exchange of materials with omentin, the blood in the vessels in the omentin, so that the cells will here function, liver function as a liver functions. And actually, this idea has been realized by Daniel Dahawan in King's College in London. So he isolated human hepatocytes encapsulated with alginate beads. So here's alginate hepatocyte beads, and he transplanted these beads to the acute liver kids. So in King's College, he's a pediatric doctor. So he performed 8 transplantations, 4 children fully recovered, 3 bridged to liver transplantation, 1 died. This is a really good data because in his criteria, most of the acute liver patient, liver kids, Philippine kids, they will die actually in his hand. So when we read this paper, they are very excited. We're saying maybe we can use our liver organoids, then we put them encapsulated, then treat the liver failure patients, right? So this is basically the idea. Then we performed the first approved concept experiment in mouse. In the mouse, we induced liver failure using extended partial We also induced the liver failure using acetoaminophene, but I'll show you only the data from extended partial In this case, we cut 85% of the liver. So the animals will die. In our protocol, we'll keep them all die within a couple of days. Then we treat them with encapsulated ELO liver organoids. You see like 2/3 of them survived. We use different donors for the organoids. We take also primary mouse hepatocytes. We didn't take a human because mouse, I think to treat mouse is much better from human because they are same species. We also tried mesenchymal stromal cells, but mesenchymal stromal cells apparently they didn't work anything in this encapsulated technique. So far, we have kind of excited with this. Then we team up with Daniel Dahawan. He's very happy to join together, work on a clinical trial. And we also, together with Xia Qiang, he's the president of Shanghai Renji Hospital. They have the largest pediatric liver transplantation center. So we're together, 3 teams we are trying to ELIT in liver failure patients. So we have tried 4 patients so far. I don't show you the data this time, but so we are still going on. The first one is for safety mainly. Then we will see primary efficacy. So with this, I would stop. So I will thank a lot of collaborators, really long time. Many people have collaborated for a long time, many years, and also my team and the money we get from. Thank you for your attention, and happy to take questions.
Very nice talk. Quick question. When you've done the encapsulated organoids into the portal vein experimentally.
No, the portal vein, intraperitoneally.
Oh, okay, because you showed a picture of something in the portal vein.
That's misleading, a little bit misleading.
Okay, so
that's what we want to do, but the way I explained, I didn't manage this, so I want to switch.
That was the question I wanted to ask. I wanted to ask if you had portal vein thrombosis as a complication, but if
Yeah,
you're
yeah,
not-
for sure. If you transplant organoid through
Yeah.
portal vein, you'll get thrombosis
Thrombosis,
for
yeah,
sure.
okay.
Yeah, yeah.
Thank you.
Hi, Dr. Hui. Excellent talk. I'm curious about your experience of using Wnt3-mediated proliferation for fetal versus adult hepatocytes and cryopreserved versus fresh, for example.
So nowadays we don't culture fetal hepatocytes because there's always a kind of ethical issues in China to using fetal tissue culture. So we try to use two types of hepatocytes. One is adult hepatocyte, one is pediatric donor-derived hepatocytes. So what we see is very clear. Pediatric hepatocytes, we can culture almost every donor. So we get them, like 90% of them, we can expand them in this medium. For adult, we can culture 1 in 2 or 1 in 3 or 1 in 4. So that's something what we have in the lab. So I think there's a big difference. I still don't know. So I told my people in the lab, I think that's very important biological question, because if you see this very clear, every time you see it, reproducible phenotype. So we don't know answer, but that's what we have observed. For freeze and thaw, I think these cells are much more stable than the fresh hepatocytes. We have tried many times, so this freeze and thaw are kind of okay in our hands. Yeah.
One quick question on safety. Do you see any chromosomal abnormalities after 9 passages of OEHH?
Okay, chromosome instability, right? The changes ultimate. Yeah, so for the passage to 8, 9, I think they are kind of okay. Yeah, so we are now expanding them into passage 18 or 20, or these things. We are still characterizing this. I think that will be a really like very important issue for the future of cell therapy. So I talked to many people in the iPS-derived cell. Many people has not think too much about the chromosome instability. They always claim like chromosome or genome are stable in their hands. But I was always asking like, if you want to transplant the cell to patient, if they are in long culture in vitro, if they are large quantity, we should characterize this more carefully. Yeah.
Thank you. I was wondering about the survival of the cells, the survival of the encapsulated hepatocyte. So what do you think? So how long can they survive? So what kind of application?
Yeah. So in vitro, they if you form this organoids, they stay really long. Like we can culture them. One of the postdoctors in the lab, she cultured them for a couple of months. Then she stopped. It's boring. She said a couple of months. So very stable as organoids. But in vivo, it's not different. So in vivo, we can keep them for 2 weeks or something. Then they die, or we still don't know what happens to these cells in vivo. Yeah.
In vivo, you would expect to treat acutely.
Yeah. So all the doctors suggest us the acute liver failure, because this is something you see clearly the therapeutic effect. So we will probably, so the first trial now in the Shanghai, this IIT, those doctors, they want to use chronic on liver, acute on chronic liver failure, because the reason is these patients are more safe compared to the chronic liver failure they choose, because these patients are more stable compared to the acute liver failure. So they want to first look at the safety. So they pick up these patients to see the safety, and then they will move to acute liver failure. Yeah.
Okay, so now we have our last micro talk for this section. This will be by Dr. Prashanth Nagesh, and he'll tell us a little bit about therapeutic inhibition of Burton's tyrosine kinase, acts on both immune cells and hepatocytes to attenuate immune cell activation and steatosis in a mouse model of alcoholic-associated hepatitis. Thank you.
Excessive alcohol use leads to development of alcohol-associated liver disease, which is characterized by increase in immune cell infiltration, leading to inflammation and liver damage. It's important to note that there is no effective therapy for patients with alcohol-associated hepatitis. So in the pathology of disease, neutrophils play a very important role. So we isolated the neutrophils from the patients and performed RNA-seq, and we found the expression of a molecule known as Bruton's tyrosine kinase, or BTK, was significantly upregulated in the patient neutrophils. We also found that the phosphorylation of these neutrophils was also significantly higher in the patient and also in the liver from human and mice with alcoholic hepatitis. So what exactly is Bruton's tyrosine kinase doing? So it's been, the role of Bruton tyrosine kinase has been implicated in metabolic disease and also severe inflammatory disease. It can regulate steatosis and inflammation. However, its role in alcoholic-associated hepatitis is not known. It's important also to note here that there is FDA-approved drug clinically used for treating patients with myeloid cell lymphoma using a BTK inhibitor. So since BTK is implicated in regulating steatosis and regulating neutrophil and macrophage-mediated inflammation, we hypothesized that targeting BTK could be a novel approach to regulate alcohol-induced inflammation and steatosis. So to start with, we looked at the phosphorylation of BTK in 3 important cell types in our context here. So the primary human hepatocytes, in macrophages, and in neutrophils, and all the 3 cell types, we found alcohol directly induced increased activation of BTK phosphorylation. Also, we found that the lipid droplets accumulation induced by alcohol in vitro was reduced in the BTK deficient hepatocytes. And also in LRP inflammasome, which is a key mediator of alcohol-induced inflammation, both in mice and humans, was shown to interact with BTK in the alcohol-treated macrophages. And other key feature was the neutrophil extracellular traps, which was induced by alcohol, was significantly reduced in the presence of BTK inhibitor. Next, we moved on to validate our findings in vitro, in vivo, and what we found was that the alcohol-induced liver damage and alcohol-induced monocyte and neutrophil infiltration was significantly reduced in the presence of BTK inhibitor in a mouse model of alcoholic hepatitis. And also importantly, the liver triglyceride level, which had increased in the alcoholic hepatitis mice, was reduced in the presence of the BTK inhibitor. And most importantly, we found that IL-1 beta as a readout of inflammasome activation, and citrulinated histone levels, which is a readout of a netosis marker or neutrophil extracellular trap marker, was increased in the alcohol-fed mice, which was significantly reduced in the presence of the BTK inhibitor. So in summary, we found a cell-specific activation of BTK in hepatocytes, macrophages, and neutrophils. And when we inhibited BTK activation using BTK inhibitor, we saw a potent reduction in features of steatosis and macrophage and neutrophil inflammation. Thus, we believe that deep understanding of this molecule will help us to understand its therapeutic potential more in detail. Thank you.
It's a pleasure to introduce Dr. Christine Knoss. She is the Whitehead Career Development Professor in the MIT Department of Biology and Koch Institute for Integrative Cancer Research. She received her bachelor's in biology from Duke University in 2010, and then enrolled in Harvard and MIT MD PhD program, where she earned a PhD through MIT Department of Biology and in 2016, and MD through Harvard MIT Division of Health Science and Technology in 2018. She conducted her doctoral research in the laboratory of Angelica Amon, where she developed tools to characterize large-scale somatic copy of number alteration in mammalian tissue, and then use diverse approach to reveal the importance of tissue architecture for chromosome segregation fidelity in epithelia. In 2018, she established her laboratory as a Whitehead Fellow at the Whitehead Institute for Biomedical Research and was honored with NIH Director's Early Independence Award. In July 2021, she joined MIT Department of Biology and Koch Institute for Integrative Cancer Research as assistant professor. Her lab develops high-throughput functional genomics approach to understand and modulate organ injury and regeneration. Welcome, Dr. Christine Knoss.
Thank you for that introduction. And first, thank you so much to Wolfram and Sangeeta for putting together yet another amazing annual gathering for the liver community. I always look forward to this. And so today I'm excited to tell you about an ongoing project in our lab that's aimed at understanding how the organism monitors the liver's functional status to properly time the amazing regenerative capacity of this organ. So the fundamental question that motivates all of the work in our lab is what underlies the unique regenerative capacity of certain tissues in the body, and can we actually confer regenerative capacity to other tissues in the setting of disease? And so if we just step back and think broadly about the regenerative capacity of different tissues, we can classify most into two extremes. There are those organs like the intestine, skin, and hematopoietic system that can continuously renew owing to the presence of stem cells that are continuously replenishing the specialized cells responsible for organ function. And then at the other extreme are organs like the heart, brain, and pancreas that cannot renew because they lack stem cells and are comprised entirely of permanently arrested cells. And the problem with this type of an arrangement is that if those cells are ever lost, there is no means of replacing them, and organ function could be forever impaired. And indeed, it's this loss of permanently arrested cells in non-renewing tissues that underlies the lifelong morbidity of numerous disease states: heart attack, stroke, neurodegeneration, and diabetes. And so a longstanding but still very elusive goal in regenerative medicine is to somehow enable the endogenous regeneration of these organs that lack stem cell populations. And so with that in mind, what's really exciting to us and what is now well appreciated by this audience is that there already exists within all of us a very remarkable and informative exception to this, and that is the conditional renewal capacity of our liver. So the liver is comprised predominantly of differentiated hepatocytes, and at baseline in the organ, there is very low turnover among the hepatocytes. However, if the liver is ever injured, those hepatocytes will immediately re-enter the cell cycle and regenerate the organ. And so this regeneration is driven not by a stem cell population, but rather by the proliferation of these differentiated hepatocytes. And so we consider these hepatocytes to be quiescent rather than permanently arrested. And so collectively, this unique ability of the liver to do this tells us that cell cycle arrest is not an obligate consequence of differentiation, and therefore organ regeneration is possible without a stem cell population. And so we have within the liver the instructions for how we might enable the endogenous regeneration of organs that lack stem cell populations. And so despite the fact that we've known about the liver's ability to do this for decades, we still don't have a molecular explanation for why it is that a differentiated hepatocyte can proliferate and therefore the liver can regenerate, while the vast majority of other differentiated cell types and the organs in which they reside are unable to do so. And so the goal of our lab is to shine molecular insight into this uniquely permissive context that we have for regeneration in the liver, so we can not only modulate regeneration in the liver in the setting of liver injury, but also ultimately identify and surmount barriers to regeneration in these other tissues. And so to do that, we're uniting two parallel but complementary approaches. The first is to establish tools for high throughput functional genomics directly within the living mouse so that we can attack these problems with the tractability once restricted to cell culture systems. And then the second is to leverage these technologies alongside what other methods might be appropriate to ultimately identify the molecular rules that are governing these permissive and restrictive contexts for regeneration. And so if we think about how the liver regenerates, this regenerative capacity is conserved across vertebrates, which means we can readily study it in mice in the lab. And the most frequent way in which we induce liver regeneration in mice is by going in and surgically resecting 2/3 of the liver in a procedure called a partial hepatectomy. Within 24 hours after a partial hepatectomy, essentially all hepatocytes in that remaining liver will re-enter the cell cycle. And over the next several days, they will proliferate and circumferentially expand that remaining liver mass, such that by 1 week after surgery, liver size and function is essentially completely restored to its original value. Now, for the viability of the organism, it is essential that regeneration occur always when necessary and never when unnecessary. Said another way, this means that knowing when to start this process and critically when to stop this process. Now, this in turn requires that the organism have a way of sensing when the liver is insufficient and needs to start regeneration. And then moreover, when sufficiency has been restored and it's time to stop regeneration. And we have no idea how the organism does this for the liver, and this is the basis for the project I'll be telling you about today. Now, when we think about the sufficiency of an organ, we can think about it in terms of its function or its size. Now, given what we know about the liver and other organs, we assume that size and function are correlated. Moreover, we assume that what matters most to an organism is not the absolute size of an organ, but rather whether it is functional. And so you can imagine that for each organ, there's an optimal set point of organ function that then leads to an optimal organ size. And so for the case of liver, if size were ever to be reduced, as would occur in a partial hepatectomy, that would manifest as a functional deficit, which would stimulate growth or regeneration to restore the set point. Moreover, if size were ever to be increased, this would manifest as functional excess that would again be detected and stimulate shrinkage of the organ. And so from here on out, when I refer to the sufficiency of the organ, I'm referring to its function, but of course, function is intimately tied to and often most often manifests as the organ's size. Now, in terms of what we know about liver sufficiency and the regulation of it, we do have compelling evidence for the existence of a set point. Across mammals, within each species, there's a very defined ratio of liver weight relative to body weight. For example, in humans, liver weight is always about 2% of body weight, and in mice, it's about 4%. Moreover, we have evidence that this set point is bidirectionally regulated. So for example, when a liver from a smaller individual is transplanted into a larger individual, creating a small for size transplant, that transplant will actually grow until it reaches the appropriate size for the recipient. Shown here is data from dogs, but this is also known to occur in humans. And in fact, in humans, it serves as the basis for living donor liver transplantation. Conversely, when a liver from a larger individual is transplanted into a smaller individual, creating a large for size transplant, that liver will shrink to reach the appropriate size for the recipient. Again, shown here is data for dogs, but this has also been shown in other mammals. And so collectively, these data suggest that there is compelling evidence for bidirectional regulation of this set point. Now, in terms of what might regulate and modulate and sense this set point, HGF perhaps is one of the most well-established roles, hepatocyte growth factor, which signals through the MET tyrosine kinase receptor. HGF was first discovered by virtue of its increasing in the bloodstream after partial hepatectomy. And indeed, it is now established that after various forms of liver injury, HGF increases in the bloodstream. And critically, this increase is driven by upregulation of HGF at the mRNA level in the liver. It is also known that HGF signaling through its receptor MET is necessary for liver regeneration. You can see here that when hepatocytes are knocked out for the HGF receptor MET, these hepatocytes are unable to proliferate after partial hepatectomy. And indeed, these MET knockout livers have impaired restoration of liver mass. By extension, hepatocyte growth factor is also known to be sufficient for hepatocyte proliferation and liver growth. I'm showing you here an experiment in which researchers infused continuously HGF through the mouse portal vein. And over that 5-day infusion, the animals that received HGF had a doubling in liver size that was driven by hepatocyte proliferation. And so collectively, these experiments substantiate a critical role for HGF in modulating liver size. However, we have no idea how the organism interprets the liver's functional status to regulate HGF expression. And so Cristina Lopez, a graduate student in the lab, decided to tackle this question. And in terms of how we're approaching this question, we first know that liver injuries of various kinds, whether it be a partial hepatectomy that cleanly removes hepatocytes or toxic injury that actually kills off hepatocytes, all result in the upregulation of HGF. And so we assume that what the organism is sensing is not necessarily liver inflammation or hepatocyte death, but rather the decline in liver function that accompanies both of these insults. So we therefore assume that a decline in liver function will alter the levels of the liver's metabolic products, and these changes will either directly or indirectly then signal to some cell type to lead to HGF upregulation. And so to understand how this sensing happens, we can simply work backward, first by identifying the cellular source of HGF, and then ultimately the signal that cell is responding to. And so Cristina began this project by first performing an organism-wide assessment of HGF expression after liver injury. And she found, not surprisingly, consistent with prior data, that the bulk of HGF upregulation was occurring in the liver. And so the next question was, what cell type in the liver is responsible for this? And so as many of you know, in the liver, hepatocytes indeed comprise the bulk of cellular mass. However, they're supported by a variety of non-parenchymal cells. This includes the stellate cells, which serve as the liver's fibroblasts, the endothelial cells that line the sinusoids, and then the Kupffer cells, which serve as the liver's macrophages. And so to determine which cell in the liver was responsible for expressing HGF, Tina went ahead and performed either sham surgeries or partial hepatectomies on mice. And then 12 hours after surgery, she harvested the livers and performed RNA FISH for cell type specific markers, as well as HGF. And as you can see in these images, what she found was that HGF was expressed essentially exclusively and upregulated exclusively by the stellate cells in the liver. This RNA FISH data is now quantified here, where you can again appreciate this near exclusive expression and upregulation of HGF by the stellate cells. Now, of course, Tina's next question was, is this a unique response to partial hepatectomy, or does this represent a universal response to liver insufficiency? And so to test that, she induced liver injury by an orthogonal mechanism by injecting the hepatic toxin carbon tetrachloride. And so you can see now in this RNA FISH data from livers injected with either vehicle or carbon tetrachloride, again, it is the stellate cells that are uniquely expressing and upregulating HGF. And so the stellate cells seem to be serving as a sensor of liver insufficiency and a key node in this sensing process. And so we can now focus on those cells and work backwards to understand how insufficiency is sensed. And so Tina's first question was, are they sensing something systemic that's circulating in the bloodstream, or is it a local signal within the liver? And so to test this, she turned to parabiosis. Parabiosis involves surgically fusing two animals along the midline by creating an excision that extends from the elbow to the knee. And in the process of creating this incision, it induces neovascularization that essentially communicates the bloodstream between the two animals. And so to test whether stellate cells were sensing a systemic signal, Tina performed parabiosis surgeries on several pairs of mice. She then waited 4 weeks to allow for neovascularization and communication of the circulation to occur. And then at 4 weeks, she went and performed either a sham surgery or a partial hepatectomy on just one of the animals in every pair, which we refer to as the perturbed animal. And then at 24 hours, she harvested both liver and blood from the perturbed animal as well as its partner. And so her first question is always, was circulation actually communicated? And to do this, she relies on the knowledge that the liver enzyme ALT increases in the bloodstream after partial hepatectomy. And indeed, you can appreciate that here in the hepatectomized animals relative to the sham animals. But critically, what you also see is that the partners of the hepatectomized animals have also increased levels of ALT, which we assume reflects ALT moving over from the hepatectomized animals, confirming that circulation is indeed shared. And so to then ask whether stellate cells were able to sense a systemic signal of liver insufficiency, Tina then performed RNA FISH for HGF and a stellate cell marker on all of these livers. And again, you can appreciate in the hepatectomized animals, stellate cells have increased expression of HGF relative to sham. But critically, also so too do the partners of the hepatectomized animals. And so the fact that the partner of the hepatectomized animal is also able to upregulate HGF indicates that the stellate cells in that partner must be sensing a systemic signal of liver insufficiency that's traveling over from the hepatectomized animal to its partner. And so this knowledge that stellate cells must be sensing a systemic signal of liver insufficiency inspired Tina to try to reconstitute this signaling ex vivo so that she could leverage classical biochemistry to try to isolate and identify this signal. And so to do this, she set up the following experimental system. She first performs either sham surgeries or partial hepatectomies on cohorts of mice. And then after 12 hours, she harvests and pools the plasma from these two groups. And at the same time that she's harvesting and pooling the plasma, she isolates non-parenchymal cells from another separate unperturbed animal. This non-parenchymal cell is a mixture of stellate cells, Kupffer cells, and endothelial cells, and she isolates this as a mixture as it allows her to isolate these cells much more rapidly. She then incubates these cells in plasma from either group for 3 hours, and then at the end of 3 hours, performs RNA FISH for a stellate cell marker in HGF. And so her first question was whether this signaling could actually be reconstituted ex vivo. And so to do this, she incubated these non-parenchymal cells in plasma from either sham surgery mice, hepatectomized mice, unperturbed mice, or PBS. And what she found was that reassuringly, cells cultured in the hepatectomized plasma had increased expression of HGF relative to sham plasma, and this is exclusively in the stellate cells. Perhaps not surprisingly, she found that cells cultured in unperturbed plasma had similar expression to that of sham animals. But interestingly, she found that when the cells were cultured in PBS, the stellate cells were expressing HGF at levels similar to that of hepatectomized animals. And so this suggests that actually the baseline and default state of stellate cells is to have high levels of HGF. And that's something about liver function, and the plasma of a mouse with a functioning liver is suppressing this expression. And so this is great. We know that the signal is something in the plasma. The problem is that plasma contains thousands of different proteins, lipids, metabolites, and ions. And so our search space is incredibly vast. And so Tina's next question was, well, is this a protein? And so to ask this question, she harvested plasma from sham and hepatectomized animals, and then she treated each of those pools of plasma with either nothing, a vehicle control, or proteinase K. And what she found was that when she degraded all of the proteins in the plasma, the HGF expression in sham animals had now risen to match that of hepatectomized animals. And so this suggests that it's a protein in the plasma of an animal with a functioning liver that's serving to suppress HGF expression in the stellate cells. Now, it would of course be great if we could just move forward and do proteomics on the plasma and just look for proteins that are decreasing in the setting of liver injury, and those would be our candidates. The problem is that plasma proteome contains 3,000 or so different proteins that span orders of magnitude ranging about a matter of 12. And so we need to somehow reduce the complexity of the search space. So just to illustrate this a bit, albumin made by the liver accounts for 50% of plasma protein mass. Another 21% or 21 proteins comprise the remaining 49%. And then this remaining 1% of the plasma proteome weight is comprised of thousands of different proteins. And so we need to reduce the complexity of our search space. Now, given that these plasma proteins have a wide range of molecular weights, we reasoned that perhaps we could use size exclusion chromatography to reduce the complexity of the system. And so to do this, Tina went ahead and she isolated plasma from unperturbed mice. And she then set aside an aliquot of that plasma that she didn't bother at all. And then she went and performed size exclusion chromatography and collected individual fractions of the remaining unperturbed plasma. And she then pooled those fractions into two pools. Fractions 39 through 50 corresponding to high molecular weight proteins, and fractions 51 to 62 corresponding to lower molecular weight proteins. And she then cultured the cells in these two pooled fractions as well as the unfractionated plasma. And what she found was that when she did that, the high molecular weight proteins were no longer able to suppress HGF expression, while the lower molecular weight proteins were still able to. So that suggests that the suppressive activity is in the low molecular weight pool. Now, if you look at the Kumasi gels for these fractions, you cannot help but recognize this major band here in the low molecular weight pool, and that is indeed albumin, the most abundant protein in the mouse plasma. And so we thought we absolutely had to test whether it was albumin itself that was responsible for suppressing HGF expression. And so to test this, Tina went ahead and performed the exact same experiment I just described to you, but this time she immunodepleted albumin from the plasma. And so as you can see, when she immunodepleted albumin from the low molecular weight pool, the stellate cells were now upregulating HGF, to a level equivalent to that of PBS. And so this indicates that albumin is actually required to suppress HGF expression in the stellate cells. Now, this immediately leads to this elegant, perhaps very simple hypothesis, which is that, well, albumin is produced by liver, so perhaps in the setting of liver function, there's high levels of albumin that suppresses HGF expression. When the liver is injured, albumin levels go down, and that relieves this suppression. The problem is that what Tina has found is that after an extreme hepatectomy, the circulating levels of albumin and the amount of albumin in the plasma is actually not different from that of an unperturbed animal. There's some regulatory mechanism in place. And indeed, she has further done experiments to show that when she reduces the concentration of albumin by half in unperturbed plasma, it is insufficient to relieve this suppression. And so this tells us that it's not the concentration of albumin per se, but some modification of albumin that's changing in the setting of liver function that must be communicating the liver's functional status and modulating HGF expression. And so just to think about it, how this might be, in terms of what albumin does for the animal, albumin really has two critical functions. First is by virtue of its high concentration in the plasma, it serves to maintain the oncotic pressure of the bloodstream. And then secondly, by virtue of its ability to bind a variety of small molecules, it serves to bind and carry a variety of endogenous and exogenous substances that are otherwise insoluble in the bloodstream. And so we suspect that albumin is binding one of these molecules, and that the ability of albumin to suppress HGF expression in the setting of functional liver is due to one of these molecules that it carries being differentially associated with albumin having signaling capacity in the setting of liver function. And so, as I had mentioned, this is still a work in progress, and that is the status of where we are thus far. What I've showed you so far is Tina has established that it is stellate cells that are upregulating HGF in the setting of liver insufficiency. And moreover, that is actually the default state of stellate cells to express high levels of HGF. And when the liver is functional, this expression is actually suppressed by some albumin-dependent signal that we have yet to identify. And so moving forward, Tina is really excited to leverage a variety of biochemistry approaches, including dialysis, to start to define the nature of this molecular signal so that we can move forward with a specific mass spec approach and identify potential candidates. And at the same time, in parallel, she's starting to dive into the stellate cells and do experiments like single-cell RNA and attack sequencing to characterize the downstream signaling from whatever this receptor is to the HGF locus. And so to end, I just want to zoom out to the big picture and bring us back to where we began, which is that by understanding the mechanism by which the liver's functional status is monitored by the organism to both initiate and stop regeneration, We hope that in time, as we continue to work on these problems, we'll ultimately be able to manipulate the gas in the brakes and drive regeneration via the administration simply of these exogenous signals. And with the growing advent of ex vivo perfusion systems like we heard about in the prior talk, this gives us the capacity to then dope in signals of liver function and ultimately drive liver growth or shrinkage on demand. And of course, this could be incredibly powerful for things like increasing the availability of donor liver tissue. And so that's everything for today. I again want to acknowledge that all of the work I presented today was spearheaded by Tino Lopez, a graduate student in the lab, and I admire so much how fearless and driven she's been in trying the variety of different approaches that this project has necessitated. I also want to thank the other incredible members of my lab who have been so enthusiastic and taking on new directions that I unfortunately couldn't talk about today. They make it a joy to show up to lab every day. We've benefited tremendously from the amazing core facilities at the Koch Institute and of course funding. And thank you all for your attention, and I'd be happy to answer any questions.
Kristen, that's really fantastically exciting work.
Thank you.
And, you know, it's interesting that the question that you begin with, what determines the size of the liver and so forth, of course, has been around for, God knows, hundreds of years. But this seems to be like the most exciting approach to understanding the question. I'm sort of curious, there are analbuminemic mice.
Yes.
Now, they're not jaundiced, and as far as I know, they don't have any other major. So presumably some other protein is carrying out whatever function is involved. And I suppose it might be interesting to see whether a similar phenomena is associated with this. Do you have any idea as to what the size of the small molecule is that's bound to albumin?
We do not. So the analbumic mice you brought up is first a great point. We've been thinking about this and we're bothered by that mouse is even viable. But we certainly are thinking about looking into that mouse for, whether there's a different carrier in that case. As to the size of this mystery signal, we do not know for sure, and at this point, we are keeping an open mind to anything from an ion all the way up to a small peptide. I think we are doing dialysis experiments where we can, of course, play with membrane size a bit, and that will hopefully allow us to narrow the search space, because we can't even really move forward with any sort of spectrometry or identification of candidates until we know the flavor of the signal. And so we hope that through dialysis and alterations in membrane size, we can actually get some handle on that. But for right now, I have no insight.
Yeah, so it's a very great talk. So may I ask you, so you showed this parabiosis.
Yeah.
So you believe like it's a secret signal for the HSC to activate HGF, right? So, but if you look at I don't know if you look at the injury like the CCL4 or something like zonation related specific injury like APAP or CCL4. So in this case, do you see like all the HSC activate are there only local activation? Is it possible like maybe there's like when you do partially or other injuries, then you have the inflammatory cytokines, they circulating everywhere. So then maybe there's a hit to the mice parabiosis animals also that they respond to this inflammation and they activate. So have you looked at carefully those activation of HSC during the injury, and also look at the activation of HSC in the how to say this parabiosis animal?
Yeah. So specifically in the setting of toxic injury, is that what you're asking?
Yeah.
So
you can choose, let's say, CCL4, which only injures the central vein area. The central vein area. Yeah. Also, you use APAP or something like this. And DDC is injure the portal vein area. So I will look at those.
Yeah. So we, in the initial RNA fish experiments, we did look at carbon tetrachloride as our toxic injury model. We did not see at face value any gross zonation of stellate cell HDF expression. There's some emerging single cell RNA-seq papers that suggest that there is some zonation on that axis, maybe that influences HDF expression. Now, in terms of the parabiosis model, we were restricted to partial hepatectomy in that model because it's the only injury model that allows us to isolate injury in the one animal, because anything else that we might inject could end up in both. But for the now downstream experiments, now that we're outside of the mouse, we can certainly test whether the plasma of a toxic injury has the same effect. And we will certainly do that as we get a better handle on what the signal is. In terms of whether there's an immune-mediated component, we don't know yet. As I mentioned, the population in our ex vivo assay is mixed. And so there could be a secondary messenger coming from Kupffer cell. And so we're now working to actually isolate the stellate cells exclusively to test that specific question.
Okay, thank you.
Hello. Yeah, it's great work. It's exciting progress, work in progress, of course. But I think, this albumin, if this is a suppressive signal, right? So it has to be, because it's so abundant, it has to be something together with this albumin or modification for the albumin has to be modify almost all of the albumin to be able to de-repress this. Right, so this must be a very abundant signal. So I know this is exciting work in progress, but I just want to say this is very exciting. It's very interesting.
Thank you. Yes, and certainly in order to, and we've thought about this a lot. You have to suddenly modify this massive population in a relatively short time interval. Now, certainly there are things that are going to accumulate in the bloodstream that are known to associate with albumin, things like bilirubin and bile acids. And so, whether it's something that's on albumin or gets kicked off due to competitive inhibition, I think there's a lot of potential ways in which you could modify the albumin via a small molecule binder.
So because you can immunodeplete, can you immunopurify this albumin from different sources and add back to see whether they still bind or do they?
Yeah, we've been wanting to do that. It is hard to immunopurify albumin in the quantities that you would need to sort of mimic plasma, but we are now looking towards recombinant albumin and whether we can sort of redress albumin with the appropriate signals.
That was really fascinating, Kristen. Congratulations. So, not to nerd it out with the previous comment, but so there's not only an albuminic mice, but people. And albuminemia is a rare condition, and those patients actually make it totally fine through the neonatal period and into adulthood, and they have other proteins that provide you oncotic pressure that we always think is, also an important function. And so, my question is, with that being said, and also obviously patients who have cirrhosis who may only have a third or half of their normal albumin concentration, does it really have to be a very prevalent molecule? Because it could also be a very rare one, and albumin is just a mere transporter and can be maybe replaced, and where then maybe minute changes in concentration, and thinking not about as much albumin modification as in just like, this is just a shuttle to get it to the sinusoids, which brings me to the second question. So if HSCs are the sensors, how do you so albumin cannot cross the endothelial space, the liver sinusoids, right? So it has to be like something gets delivered and then diffuses in. How do you get it to this stellate cell?
Yes. So I think in terms of the abundance of the signal. So in Tina's experiments where she does fractionation, she ends up dividing the protein amount, or at least by a factor of 3 in some of her experiments, and that does not relieve suppression. And so it is consistent with this possibility that it is not all albumin molecules that necessarily need to be signaling in this way, but some signal that is dependent on albumin, and it doesn't have to be particularly abundant. Now, and we are certainly entertaining the possibility that it is merely albumin is just allowing it to even stay in the bloodstream and signal. Would albumin be able to pass through the fenestrations though to signal direct? Okay. So I guess I had imagined they could pass through just in the way that viruses do, but perhaps it gets close enough and then it allows for transport across the cell. Or alternatively, there is a second messenger that is getting captured in our ex vivo system where there is a, maybe the endothelial cells are then communicating to the next one.
I would like to ask a couple of questions. One, a quick one. Do you see any difference between plasma and serum?
No. So we initially worked with serum and we had the same effects.
And then it's interesting how you actually clear the albumin from that higher molecular weight fraction. You use an antibody?
Yes.
And if so, you could do a mass spec analysis of the albumin before and after partial hepatectomy and time course. You really can have a really good tool to find that small molecule you're missing.
Yes, so we do, we immunodeplete albumin for that assay via an antibody on beads. In terms of moving forward with mass spec, we don't know whether we should be doing small molecule mass spec or protein mass spec yet. And so that's the key question we're trying to distinguish now before we would move forward. But that would absolutely be the ideal approach, is we pull down the albumin with whatever is associated and then send it down whatever is the appropriate mass spec pipeline. But right now we don't know which mass spec road we should go down.
Thank you, Dr. Knoss. We can
Thank you.
save question for reception.
Okay, excellent. Thank you so much.
All right, so thank you everyone for staying. My name is Carol Alcedo. I'm an assistant professor at Auburn University, and I'm your co-chair today for this session. So I'm excited to introduce our last speaker of the symposium, Dr. Miriam Merad. She's the chair of the Department of Immunology and Immunotherapy at Mount Sinai School of Medicine. She's a leader in the fields of innate immune cells, specifically dendritic and macrophages. And she has identified lineages of tissue-resident macrophages as well as dendritic cells and their roles in pathology or in cancer progression. She has founded and is the director of the Mark and Jennifer Lipschultz Precision Immunology Institute at Mount Sinai. And this really brings immunology discoveries to the clinic. She's also the director of the Mount Sinai Human Immune Monitoring Center, which uses cutting-edge single-cell technology to understand the contribution of immune cells to major human diseases or treatment responses. She's an elected member of the American Society of Clinical Investigation and the recipient for William B. Culley Award for Distinguished Research in Basic and Tumor Immunology. And finally, she's currently the president of the International Union of Immunological Societies and has been and was elected in 2020 to the National Academy of Science. Let's welcome Dr. Merad.
So it's a pleasure to be here. I'm sorry I couldn't be with you, but I had to stay for fundraising purposes, and I would rather be talking science with you guys. So I'm not sure how much the audience knows about immunology, and it's difficult to discuss virtually. So, but still, if it's too complicated, say something, and I'll stop and I'll answer it. So I'm going to talk about immune response to cancer, to liver cancer, and how we are really trying to understand that response to develop novel immunotherapeutic for liver cancer. Now, before I dive into some of these findings that we've made, I just want to remind the audience that most adult tumor, whether it's in the liver or any organ, contain an inflammatory component that shapes disease outcome. Right? The goal of the immune system, or the the mission of an immune response of the immune system is to constantly survey for threat and respond to them, respond to the threat, mostly to eliminate that threat and respond to it by producing immune cells, often in activated state, that will go to the site of injury and try to delete, repair, etc. And this is what we call inflammation. So in any type of disease where there is an injury, you will find an inflammatory component. And the moonshot challenge that we have as cancer immunologists is to rewrite the code of that immune response to enhance anti-tumor immunity without inducing tissue damage. So let me explain this just for a minute, because it has reshaped the way we think about cancer therapeutics. So clearly, cancer is a dysregulated cell type that starts proliferating and metastasizing, and it's been difficult to target that dysregulated component. And we realize that the immune response against these cells that are damaged, because that immune system is not dysregulated, is not malignant, can be used to eliminate those cancer cells, the same way we use the immune systems to eliminate any infectious cells. Cancer cells have many types of mutations that are recognized as foreign. The immune system can come and eliminate. The problem is that the amount of foreign antigen that's expressed on cancer cells does not always allow the immune system to distinguish them from the tissue, so it can also damage the tissue. And also the tumor find a way of dampening that immune response, right? So the challenge that we have is how to reawaken, make sure that the immune system is not fooled by the tumor and continue to attack it, but it attack it specifically without attacking the rest of the tissue. And I think now we believe as a group, as a community, that if we found a way to specifically reactivate the immune system, specifically in the tumor lesions, without affecting the adjacent tissue, we would be able to eliminate most cancer lesions. The problem is we still do not have a way of unleashing an immune response in a very specific manner. So to really understand first the cellular composition and molecular composition of the immune system, but also the tumor cells in any lesions, we built this group now a few years ago, which we call the Neoadjuvant Research Group to Evaluate Therapeutic. And this group's mission is to find informative tissue that we can study at the deep molecular and cellular level, at single cell level in particular, to start learning about the program that are induced in response to tumor cues. Okay. And here we decided to spend lots of time using this window of opportunities where patients are priors are recently diagnosed, so we will have access then to treatment-naive lesions, and then we can use that are resectable. So we are focusing on small lesions, treatment-naive and resectable, so that we have the whole surgical piece to study. But also prior to surgery, we can then expose the patients to perturbation, so drugs, immunotherapy or combination therapy, and that will enable us to really study molecular response to immunotherapeutics. And we think this is an essential it's an essential experiment. I'm going to stop because I think I am on the setup. I just want to voilà, manually. Okay, perfect. And we think this is very important. These experiments are very important because we realize that often the clinical trial have difficulty to many clinical trials fail at late stage. So we have, interesting results in phase 1 and phase 2. We bring and we do this large study, and usually they fail to reveal a difference against the control arm. And we think that this is because we still do not lack a very good understanding of molecular response to drugs. And the more we learn about these drugs and the more we learn how patients respond to drugs, we can de-risk, this big cost that is associated with phase 3 trial. So there's two reasons why we build this framework is of window of opportunity of pre-surgical time where we can study the lesions to learn about molecular composition of a lesion, but also use that window of time to expose patients to any type of treatments of interest and learn how patients are responding. And then when we make, these hypotheses or establish this correlation, then we can hypothesize about mechanism of action and test them in your model of preference, right, in the laboratory. So we study mice quite a bit, but can study it in any preclinical model. Okay, so this is an example of a study that we've done to learn how HCC respond to checkpoint blockade. And here we took advantage of what we call this neoadjuvant trial. It's in fact a pre-surgical trial where patients with early lesions, so they were resectable lesions, will be exposed to 2 doses of checkpoint blockade. PD-1 antibody blockade is supposed to reactivate T cells. And these T cells that are reactivated have been shown to sometimes unleash a cytotoxic activity and eliminate cancer cells. So why they didn't do it previously? Because they are fooled by tumor and its microenvironment that's trying to quiet down immune response in order to enable the tumor to grow. And it fools the system because the system is thinking that it needs to repair, and then the T cells start to quiet down and work from now many investigators, but initially by Tasuku Honjo, Gordon Freeman, and Alan Sharp have shown that PD-1 is a key molecule that controls, in fact, T cell activity. And another checkpoint is CTLA-4, and discovered by, which role was discovered by Jim Allison, that also reduced T cell activity in tumor. So here we are going to say, okay, we give PD-1 blockade to patients with HCC. Are patients responding? And why do they respond or resist this treatment? Okay, so this trial was published in 2 years ago, and we included 28 HCC patients. 25 patients responded, and this enabled us to then compare molecular response versus in responder patients and resistant patients. So first, something that surprised me when I was looking at this tumor, so HCC, the HCC that we saw was mostly viral-induced HCC. We had some NASH, but it mostly was hepatitis-induced HCC, and this is the prototype inflammatory tumor, right? So I expected that all the tumor lesions will be full of T effector lymphocyte. And on the contrary, we saw the 3 patterns that have been described in many tumors, where in some tumor you have a lot of T cells. T cell is that effector lymphocyte that can eliminate tumor cells. In some cases, you see T cells that are excluded, so only in the stroma that are surrounding the tumor. And in some cases, there's very low number of T cells. Now, we know that if tumors are low in T cells, they are not going to respond to checkpoint blockade. This molecule reactivates T cells. So if there are no T cells, it doesn't work. So we wanted to focus then on this homogeneous histological group that is enriched in T cells. Okay. And this is what I'm showing you here. In fact, all responders to PD-1 blockade are T cell rich, but there is a group of patients that were enriched in T cells, this is what I'm showing here, enriched in T cells, but didn't respond. And this is what we wanted to compare, because immunologically it suggests that in both cases, the T cell, the immune system has recognized liver cancer antigen, it accumulated at the tumor site, but it was unable to kill the tumor cell in one case, and able to kill the tumor cell in another case. And this is what we wanted to compare so that we can design novel therapeutics for this resistant group. Okay, so And first things we wanted to know is whether the tumor mutation burden was different between the two groups. So the idea is that the more mutation you produce, the more foreign antigen you express at the cell surface, and the more immunogenic the tumor will be. So we wanted to know whether this was the case in HCC, and we didn't see much difference in terms of mutation, accumulation of mutation between the responder and non-responder. Definitely within the T-cell rich group, we didn't see any difference, which makes sense because, you know, that T-cell, in both cases, T-cells were accumulating, so they were seeing, you know, tumor antigen. Then we looked at the type of mutation that were associated with response on resistance, and also the type of mutation that were associated with T-cell rich and T-cell low tumor. And what was interesting is that in the T-cell low tumor, so this is non-responder T-cell low, right? This low means T-cell low, that when there is very little T-cell, there was an enrichment in this beta-catenin activated mutation. And we're very excited about this because in our Mars model that was developed by Amaya Dujambio at Mount Sinai, she had shown in a study where she collaborated with my lab, we found that this beta-catenin activated mutation was associated with reduced immunogenicity. For many reasons, the priming of T-cell doesn't work so much in these mice. And what is also interesting is that in melanoma lesions, that same mutation was also associated with resistance to immunotherapy and reduce immunogenicity and reduce accumulation of T-cells, and likely, you know, through the same mechanism. And it's important to think about this because I firmly believe that there is a lot of commonality of response of the immune systems to tumor cues, you know. So I'm an oncologist, but my cancer biologist colleagues like to focus on the tumor cells, thinking that each tumor and each mutation will really have a very different role in designing and shaping the microenvironment, and then each we should be treated differently. And although this has transformed, you know, cancer care with the discovery of some targeted mutation, we think that immunologically it is unlikely that there is so many differences. There will be big differences. So, for example, this beta-catenin activated mutation that reduce, you know, this priming, but we can then have commonality of response that are driven by the immune system just recognizing cell stress and cell death, right? So, and there is a finite way of responding to a threat for the immune system if it's not dysregulated, if it's properly working. And this is what we are trying to uncover. Okay, so are you guys following me? Or this is too immunological? If someone can say yes or no, I'd love it. If I'm too technical, someone can come to the stage. Am I very technical?
No, you're good.
I'm good. All right, thank you. All right, so now we are going to ask differences, why some patients respond and some patients didn't respond, right? So you have this T-cell infiltrate in liver cancer, we treated with PD-1 blockade, why some respond and some do not respond. So now we are going to look at the molecular program of immune cells that are infiltrating these responder and non-responder lesions. And what we saw is that the response was associated with expansion of specific molecular program within T-cell. So there was a big expansion of CD8 effector T cells, an expansion of CD4 T cells that usually is found mostly in the lymph node. And those CD4 T cells are very good at activating B cells to produce antibodies. Okay, so we saw that as being very strongly correlated with response. So that became interesting to us because now we have, we know that inducing that type of response is probably important if we want to reduce tumor progression. Now, this expansion of CD8 T cell has been largely reported upon treatment with PD-1 blockade and the big correlate of response. What was less reported is these CD4, which are not effector cells. Those are cells that provide help to CD8, can induce B-cell antibody response, are mostly found in the lymph node. So it was interesting that we're able to detect them at the tissue site. okay, we saw that this expansion was clonal. Okay, so that's important even for non-immunologists because it suggests that it was not random. The cells that we reactivated upon treatment with PD-1 blockade with this checkpoint were in fact recognizing tumor antigen. They were clonally expanded. They recognized a threat. And because these clones were present in the tumor site and not in the adjacent, we assume that they were responding to tumor cues and therefore to tumor antigen. So clonal expansion is something that's very important for immunologists because it suggests that the response is specific, and then we can then target the tumor while sparing the adjacent tissue, right? Which is the dream of immunologists, is use the preciseness of an immune response against infection, right? When you have flu infection, your immune system is eliminating the infected cells and sparing the cell next to it, which is not infected. So this is the precision that we are trying to obtain here. Okay, so for the molecular-minded in the audience, this is a beautiful experiment. So here, when a T cell expands against a tumor antigen, it has one barcode, which is this T cell receptor, which can be sequenced, at the molecular level. So each clone has exactly the same receptor. So now we can purify the T cell, sequence the part of the gene that's going to lead to that T cell receptor, and here we have a clone, right? So here, each bar is a different clone, and we are going to look at the stage in the molecular stage within the clone. So the cells are proliferating, and then the cells are, accumulating this clone, and we are going to say in what molecular state these T cells are. And what we found is that, in fact, there was all the different states of T cells that usually happen in the lymph node were occurring at the tumor lesions. So the cells were proliferating locally, differentiating into effector cells, and sometimes also differentiating into terminal cells. Now in the responder, we saw a lot of effector T cell differentiation. This is good because this is the state in which a T cell can eliminate a cancer cell. In non-responder, we saw very dysregulated differentiation, the terminal differentiation where T cells lose the ability to kill. But in all cases, we saw that there was a lot of progenitor stage. So T cells were at the progenitor level, they were proliferating locally and either differentiating toward terminal differentiation or effector differentiation. And this is something that's very interesting to us, right? Because it suggests that maybe we can change the trajectory of these T cells and make them kill the tumor cells. We've done another experiment, which is quite a tour de force, where now we are going to image these TCR sequences. We are going to image them by using a probe that can visualize the sequence of these clones. And we are going to ask, are these T cell clones, were they present prior to treatment? At least in the responders. So what we did is we designed probes against the most expanded clone, and we are going to ask, were they present at the tumor site before the treatment, or are they being recruited from the lymph node? And what we found is that all the T cells, that this is each dot is a clone, the different TCR, all the clones that were found were in fact present prior to treatment. They were already in the liver lesions. We didn't have to recruit them. We were just re-expanding them locally. Okay, we were expanding them locally. And that is very interesting because it suggests that there are local cues that may be enabling this re-expansion that we need to identify. So what we saw is that many T cells were expanded locally upon exposure to PD-1 blockade. And therefore, we started to search for local cues that promoted the expansion of these effector T cells, especially in responders, and because we didn't see this expansion in non-responders. Okay, so now we are going to focus in responder and say, well, where is this happening, right? So this PD-1 can re-expand the T cells in the middle of this liver lesion, and where is it happening? And what we saw is that most of the re-expansion occurs in these hubs that we described recently. And these were hubs where, so this is a spatial transcriptomics analysis that was using this technology called MERFISH, which uses 400 transcripts that we identify by single-cell RNA sequencing of the immune lesion. So now we ask how the immune cells are being organized locally. And what we saw is that in the responders, we saw this triad. We saw dendritic cells, these cells that my lab has been working on for quite a bit, being localized close to a CD4 helper that we saw expand quite dramatically, but also progenitor CD8, not the effector one, but the CD8 at the progenitor stage. And we saw that being very organized. And now with MERFISH, we can even know what type of molecule, inflammatory molecule, are being produced in these hubs. So that's quite exciting. So we saw these, and then we started to measure these hubs or this triad in responder and non-responders. And what we saw is that there was a high enrichment, at least a higher proximity enrichment between these cells in responders compared to non-responders. Suggesting that really this is a little hub that enabled the differentiation, we believe, progenitors into effector cells. And indeed, in gray, we saw this effector CD8 which kill the tumor. In fact, they are coming from these hubs, and then they are going to the tumor cells and eliminating them away from the hubs. Okay, so this is important. Very important, because it suggests that we identified the site of reactivation, right? And it means that this is potentially what we need to target. And one thing that we think a lot about when we think, I mentioned earlier, drugging, the tissue site. Now it suggests that what we should be thinking about is drugging this site or potentially recreating these hubs, right? So thinking again about the specificity of targeting or modulating an environment. So we then went back to spatial transcriptomics and started to look at the molecular composition of these immune hubs. And we started to see that indeed we started to look at the molecular program of the cell. So here by MERFISH and imaging, for sure, we saw that there were these dendritic cell and progenitor. Now we want to go a bit deeper, looking at the molecular program of the cells that are present in these hubs. So the spatial transcriptomics, this I think it was Visium HD, enables us to look, have a larger capture of transcript. And we saw that the dendritic cells that were localized in these hubs have a very clear a program that we had already identified and showed that this is the state in which these have eaten a cell debris and is in a state of antigen presentation activity. So DC that accumulate, the dendritic cells that accumulate in these hubs are probably charged with antigen, which makes sense, right? So they come charged with antigen, and maybe they are contributing to forming these hubs. And I just want to say something about these hubs is that a few years ago, in a different model, we do exactly the same exercise in also inflammatory diseases. And an inflammatory bowel disease, we saw exactly the same organization. And the dendritic cells that were in the midst of these hubs were also in exactly the same state of which we call a mature DC enriched in regulatory cell. This is a molecular state that we study at the molecular level in mice and in vitro, and we believe is driven by very specific cues, including lipid cues. The cell debris that is eaten by dendritic cells is full of cholesterol, and we show that cholesterol movement is contributing to the maturation of these dendritic cells. So there is recently also another paper in lupus showing again this lymphoid structure in lupus, and in the middle of this immune lymphoid aggregate, I will say, there was these dendritic cells in this exactly the same state. This BERC3 and CCR7 is one of the markers of these dendritic cells. Okay, so I wanted to talk a little bit about, well, I will finish with this. I had a few more slides, but I think I went probably over time here. But I wanted to go back. I think that there was discussion about inflammatory diseases in the liver, but I want to highlight this immune aggregate. So these immune aggregate are first, in chronic inflammatory lesion, in chronic lesion. When you have a chronic lesion, you always have you most often have, if you have a strong immune system and there was no dysregulated tissue cues, the immune system should be able to recognize the chronic lesion. So in any chronic lesion, you should have this chronic inflammatory component. And when there is chronic inflammation, there is this organization of the immune system that resembles the lymph node that happened at the tissue site. And the most mature stage of this aggregate are what we call the tertiary lymphoid structure, where you start forming a pseudo lymph node at the tissue site. And this is a big site of pathogenicity in inflammatory disease, because this is where effector T cells or antibodies are being produced. But in cancer, it's probably a stage where that can be reactivated to unleash an anti-tumor response. So a big focus of my lab is to understand how these tertiary lymphoid structures are being formed, and we are looking at the role of dendritic cells in forming this tertiary lymphoid structure. And in a study that we are about to submit, Raphael described how these dendritic cells are contributing to recruiting immune cells at the tissue site, and identify some of these molecules that contribute to the formation of this immune hub. So with this, I'm going to stop and maybe just go to my last slide that I think really has the take-home message is that these, well, I don't know whether it has a take-home, very discrete message here, but what I wanted to emphasize, I think I had a summary slide just before that, but what I wanted to emphasize is this spatial organization, the spatial organization that occur in chronic lesion, whether it's cancer or chronic inflammatory lesions, that in fact we needed to understand better. So, and using spatial technology, for example, but also single cell technology or functional spatial genomics to understand how this cellular organization are really contributing either to reactivation of the immune system, which could be pathogenic or could be beneficial. So this is something that we've started to do, where we have used now this receptor ligand analysis based on all the spatial transcriptomics and single cell transcriptomics that we've done in these hubs. And we have identified key receptor ligand molecule that we are now trying to drug to either reactivate the hub or dampen the hub in inflammatory diseases. Okay, so with this, I'm going to stop and thank my lab, Raphaël Matthews, the story that I didn't show, and Pauline Lamarche for the first part of the work. And this is work in collaboration with many different people, including clinical group. Very important to have a very close ally in clinic and pathology and in radiology. Some of the trial was funded by Regeneron, and we have several collaborators with whom we brainstorm many of the experiments that we do. All right.
We have time left for just one question.
Sorry. I did something bad. I should put my video on. Sorry, I am still here. I did a mistake with my video. Start video. Perfect.
Hello.
Yeah.
Can you hear me okay?
Yes, very well.
Okay, Luba Bartikovsky, I'm at the NCI, and I have a question to you. There has been recent renewal of attention to the B cells in the PD-L1 response. And do you have a feeling or you have any knowledge of what is special of those B cells, aside from being germinal cell, but germinal central cell, but what is there about those B cells that participate in the immune response? What do you think, where is going?
Well, first, your question reassured me because there is some immunology knowledge in the audience. I wasn't sure. So this is a great immunological question. So up to now, we thought that B cells or antibodies were not really major contributor to to tumor cell clearance, right? That's somehow of a dogma in immunology, not a dogma, but it's paradigm in immunology. Happens, so what we are now realizing, many of us, is that there is a clear correlation between those structures that I just described and response. We are also realizing there is B cell clonal expansion, germinal center formation, antibody production that correlate with responses. And in fact, in a recent study, so here I showed in this study, I focused on the T cells, but we are about to submit a paper on the B cell response. So here, where we look at the quality of the B cell and what they are recognizing in responder and non-responders. And what we are seeing is that the responders are associated with enhanced tumor-specific antibody response that are produced in these hubs and recognize tumor antigen. So many of us are now realizing that these antibodies can participate in clearance. They potentially can also provide a tumor antigen to APCs. You know, DCs and macrophage eat through this Fc gamma receptor, and they could be a way of capturing antigen and furthering, antigen presentation to T cells. So there is a lot of excitement about reanalyzing the B cell compartment and revisiting the contribution of, for us, who is mostly the antibody not B cells as cellular. It's possible they also produce inflammatory molecule and help to, and this interaction with CD4 could potentially play a role. But we are thinking mostly about B cells as either a source of antibodies, source of clearance, or tumor antigen to APCs.
Thank you, Dr. Merad, for your wonderful presentation. Now,
So I just wanted to say a word of appreciation on behalf of all of us. I'm told that over 400 people attended on site or virtual. And what started out as a birthday present 35 years ago has been one of the longest lasting ones that I've heard of. So a special thanks to Sangeeta, to Wolfram, and of course to Heather, who has done an enormous job in making all of this possible. I would like to add something, though, that the purpose of this symposium has always been to be somewhat at the crest of the wave. The big challenge is to bridge the exciting things going on in basic biology, computational sciences, engineering, with medicine and an understanding of disease. And that gap seems to get bigger and bigger and bigger as science advances and medicine gets a bit frustrated. But I'd call your attention to a part of this program that's unique. And that is Heather has put together each year the list of names of speakers over the past 33 years. And if you look over it, you will discover it's virtually a who's who of people who have done outstanding research. And there are a good number of people who were truly at the crest of the wave, and a few years later won a Nobel Prize or the National Medal for Science or election to the National Academy and so forth. So for all of you, particularly young people who presented really brilliant, exciting work at the crest of the wave, we hope that things like this will happen to all of you. So thank you all for being here, and thank you particularly for having organized this. and also to the American Liver Foundation, which really is the body that's been supportive of this venture ever since it first started. So thank you all.
And I wanted to say hello. My name is Lorraine Steele, and I'm the CEO of the American Liver Foundation. And I have been in this role for the last 4 years. And I gotta say, this program is my most favorite. I love supporting the basic sciences. And we are so grateful for Win for having, and Luba, who had the idea of the birthday party, right? All those years ago, and how that has driven so many advances. Of course, we are so appreciative of Wolfram and so appreciative of Sangeeta, wherever she is. And of course, the amazing Heather, round of applause, who just works on this all year long. And we are so personally appreciative of Heather's efforts. I did want to have a little bit of a shout out to my colleagues. One who's watching now, who's been behind the scenes, Dr. Ellen Jordan, who leads our research program. She's a PhD has a neat PhD in immunology, been very active. And let me tell you, we want to have a relationship with all of you. We want to be involved with your careers and hopefully funding it as we continue to promote research and particularly the basic sciences. So please reach out to Ellen and introduce yourself. She wasn't able to join because we had this big conference. You might have heard of it, AASLD, that we've been tied up for quite a few days. But Dr. Jordan's email, just so you know, is hjordan@liverfoundation.org. Also, shout out to Asma and Caroline and Heidi who've been involved. I just want to spend a couple of minutes talking to you about the high points of the American Liver Foundation. We are blazing new trails and stepping up. In particular, our research. We are very proud to have funded last year 10 young investigators, 3 liver scholars, which are 3-year appointments, $75,000 a year for 3 years. And then we have postdoctoral fellowship grants. And that was after COVID. We had taken a hiatus a little bit during COVID, came back strong. This year, even stronger. We have just funded 8 pilot grants in various areas of liver, and we are right now in a review process, and we hope to fund a record amount of additional grantees, hopefully 18 to 20 researchers this year. And we have just scratched the surface of what we're going to be doing moving forward. So this is a great time to be in liver and spread the news to your friends, your colleagues. We have got to entice people to get in the space because patients need it, right? We got 100 million people out there just in the US and billions worldwide. And we need all of your creativity and all of your innovative ideas to help these individuals. One last thing wanted to share. We have launched a patient registry, and it's first ever for all liver disease states, all of them. And we are getting rave reviews from the NIH and from clinical centers and physician scientists across the country. And so that will be a great resource to you moving forward. And again, thanks to Dr. Jordan for driving those efforts. I did want to acknowledge one of our board members here, Dr. Ekong. You heard from her earlier today. We also had Dr. David Cohen here for most of the day, one of our board members. We can't do it without our leadership. But bottom line is, what an event and what an inspiration Dr. Irwin Arias is to the field of liver. And I think we need one more round of applause for Win. and his leadership. I know he's been such a mentor to me. When I first came to ALF, every couple of weeks, every week actually, for about a year, Win gave me 1 hour of his time to teach me, to educate me, to push me, to encourage me. And that made a huge difference for me and also for the American Liver Foundation. So thank you, Win. One more round of applause for Win. Thank you all. Now, Heather, the real exciting stuff.
Okay, I promise we're going to be short. I will not, my children always tell me too many words, Mom, so I will try and be brief. The poster session is kind of my special project with this, and it's something that I've been so glad to see it taken up so well. And I just want to highlight, we had a completely full poster session. You've all been at meetings where often that doesn't happen. Every single judge who volunteered to judged all 4 posters. We had all of the data that came in. And what I was really pleased about is everybody who I asked to judge who judged last year were enthusiastic and donated their time again to do that again this year. So I think it is a special piece of what we do together. So our awards, we'll start with fan favorites that you all voted for, goes to Sam Morningstar. He's here, he can come down. Oh, there you are. Hello, congratulations. And then the runner-up of the judging scores goes to, oh shoot. I don't know how to pronounce your name. Wait, I got it. Teodora Jurosevic. Congratulations. And then our highest prize from the judging and also second highest in fan favorite. So it was really, truly popular, goes to Christina Lopez. Awesome job. Thank you. Yes. So now Wolfram had just a couple of things. And then as everybody else goes out to enjoy the wine reception, we want to have a picture with all the poster winners or poster presenters.
Thank you. I'm between you and the reception, and so I will be super short. Again, I want to thank the ALF and everyone who got involved and was involved there in both preparing for and running today's meeting. In addition, I want to just thank Brooks Firth Bard, who helped during the registration and the event, and then Michael Sweeney in my division, who helped with a lot of the pre-planning together with Heather. And then people here at the Broad. The Broad, I think we can't find a better place to epitomize both community and innovation than this institute. And both Mary in events and Sean in AV behind that door has really helped us greatly to put this day together. Avantor and Corning were our corporate sponsors and really helped with the funds. And then, as Heather already said, the judges, not only those, but the session chairs and obviously our 10 speakers who really enriched us and helped us see the cresting wave and the path ahead. But I really want to thank Heather one more time, because it's easy for Sangeeta and I to stand here, but it's really Heather who puts this all together, and that's fantastic. I want to thank Win for inspiring not just us here in the room, but really generations of liver researchers locally at all the places you worked at, and nationally, internationally over the course of your lifetime. And then I want to thank all of you. When we took this over, Sangeeta and I, as she said in the morning, we wanted to build community. And for all of you who come back every year, pay attention, sit through the day with us online here in the room. That is the community. And actually, that's what it takes for us to move the needle on behalf of our patients. And so that's really fantastic. And with that, I'll end. We're going to see you back next year and enjoy the reception.
We would like to take a picture of the winners of the poster with Win, and everybody who presented the poster, can you also come down? We'll take a picture with Win with you. So can you come up, Win? Come on. So 3 of you with Win.
All right, hello. 3, 2, 1. Okay, now everybody presented that was maybe some of them go in the back, the shorter people, and the taller people in the front, and we could take that. You got it. Okay, someone will come in front. All right, one in the front, all right. So everybody would say liver and smile. You can't see me, I can't see liver. All right, now make a funny poster. I mean, come on. Funny, it's not so serious. That's fun. Thank you, guys.